In this juncture, I would like to recall some of the important works that NIS has taken forth in the field of virology. At least one or two studies that NIS has taken up. We all remember the height of the pandemic, COVID pandemic in the year 2020. So that time NIS had taken up 3 or 4 very important clinical trials, particularly in COVID-19, and we have associated with SRM Medical College and Government Chengalpattu Medical College in carrying out randomized controlled clinical trials, and we had seen tremendous results, and they have been already published and it's available in the website. You can always refer to that. If you just go to the Google and type Siddha plus RCT, our clinical trial will come and fall first. So you can go through that. Not only that, we in our institute had initiated a in vitro anti-SARS-CoV-2 study in collaboration with International Center for Genetic Engineering and Biotechnology in New Delhi, and that study is also underway. The results are already come up, and we've seen very encouraging results for the Vishwasura Kudini. That has got very promising results against the SARS-CoV-2 virus. Not only that, we have the neem trees ubiquitously growing in our country. And turmeric also we use left and right, day in and day out. So we thought why we could use and we could study and use this neem and turmeric, which is very ubiquitous, because 1 kilogram of Kavasura Kudini costs around 1,000 rupees. So we thought why we go for a cheaper and a very effective alternative, cost-effective alternative, which is found very ubiquitously. So we thought like that, and we went about doing the anti-SARS-CoV-2 assay, and we found wonderful results, and we are now in the process of communicating the manuscript for publication. Not only that, we had a long association with Professor Dr. Pushkala ma'am, because my PhD was on primary dysmenorrhea, a clinical trial on that. In that study, I had to do prostaglandin F2 alpha assay in menstrual blood, and I contacted ma'am. Ma'am was the one who ran the ELISA assay for me, and I got wonderful results in that. And at the time of public viva, the then Vice Chancellor, Professor Dr. Geeta Lakshmi ma'am, congratulated me very much. A study should be like that. She was complimenting me. So I'm very grateful to you, ma'am, at this juncture. It was all because of you that work was seeing the light of the day. And today morning we had some wonderful interactions with Professor Dr. S. Saravanan from Savita Medical College. And we had some common interests, discovered some common interests. Sir was very keen and bent upon in doing the research in hepatitis viral infections, particularly hepatitis B and hepatitis C. And we were sharing like, why don't we take up a cohort study, say about 5 or 6 years, long-term study, because in our system of medicine, we've got wonderful drugs which can curb and curtail the viral infections, and not only that, our drugs are known for preventing the relegating of the state of health in the liver, those who have infected with hepatitis B and C, progressing to the level of cirrhosis and even hepatocellular carcinoma. Our drugs are wonderfully good, have been proven in animal studies, in one of the studies done at Manipal University, Karnataka, that our drugs like the Kadukai Matre and other drugs like that, they prevent such stage progressions. So we thought, why don't we take up such a study in the days to come and we have had some useful discussions this morning, and hopefully it will continue even after this seminar. Thank you so much, sir, for being here, and you are going to hear from him about the wonderful topic, Hepatitis B viral cure paradox. Really a paradox. It's a very evasive infection. So Shar is going to deal with the nitty-gritties of the infection as to how we can deal with them with our system of medicine. So all in all, this workshop is going to be a very useful one. So make the most of it, you dear students, and don't miss out anything. Take every minute detail of this. Take notes. Maybe this might seem insignificant for you at this time, but as you grow old, you'll certainly understand the importance of the important words of wisdom that's going to be given from the experts we have invited. So with these few words, I congratulate once again the organizers for having organized such a wonderful workshop, one-day workshop, and thank you for the opportunity given to me. I close my remarks. Thank you.
Dr. Manjari ma'am to kindly address the gathering.
Good morning to respected Director Professor Dr. G. Sindilvel sir, distinguished today's speakers, Professor Dr. Pushkala ma'am, Professor Dr. C. Saravanan sir, respected Dean Research in Charge, respected Madhu Ramachandran sir, and Dean, the faculties of Department of Microbiology, and all the faculties, my beloved students. Happy morning to all. It's my pleasure and honor to give felicitation today's seminar, Detect Microbes, with the theme, Current Practices in Infectious Disease Diagnostic. I congratulate Madhu Ramachandran sir and the team for choosing this seminar relevant to the timely theme. Current healthcare landscape, to diagnose and currently evaluate the infectious disease cannot overstated. Emergence of new pathogens, resistance, antimicrobial resistance, and evolving challenges for diagnosis highlights the critical role of microbiological diagnostic. First and foremost, I congratulate Madhu Ramachandran sir and the team to select this topic for today's seminar. I hope our today's experts share their insights, experience, and knowledge that strengthen our ability to detect microbes 2025. And it also reflects the National Institute of Siddha commitment to integrating with traditional knowledge to modern diagnostic advancements for holistic healthcare. I hope and I am the confident today's seminar topics will great confidence and ability to all the faculties and students and the clinical team to pave the way to collaboration and innovation of the healthcare management through microbiological diagnostic. Once again, I extend my best wishes to the Department of Microbiology, Madhu Ramachandran sir and organizing team. Great success of the seminar and extend and commend the great initiative and motivation of Department of Microbiology under the guidance of our Director, Professor G. Sandilved sir. I thank you all for this opportunity. Thank you.
Thank you, ma'am, for your encouraging words. And now request Dr. B.R. Sandil Kumar sir to address the gathering.
Hello, students. Good morning, everyone. I don't know much of that. Good morning. Good morning. Yeah, it's going to be a great learning. So both are a very interesting topic selected by the Division of Microbiology, Department of Integrative Health and Translation Research. First one is going to talk about techniques. Basically, Siddha, many of the drugs which we believe that acts to the immunomodulation, basically. So, learning the techniques is very, very important in this junction. Similarly, Manja Kamalingo, Siddha Matum, Pirike Mudiyada. So, common man always think whenever they affect with the jaundice, they think about the Siddha medicine, basically the Hepatitis B or A, B, C, or some, whatever it may be, basically. So, certainly it is going to be a great learning for you, all of you. We have our distinguished speakers here, so you have a wonderful learning today. Have a great day. Thank you for the opportunity. Thank you so much.
We take great pleasure in recognizing and congratulating the prize winners for DNA Day quiz program, held in commemoration of International DNA Day, April 2025. I now invite our Director, Professor Dr. Sandilved sir and Dr. Muthuyan sir to kindly present prizes and certificates to the winners. The first prize goes to Ms. Nimmi Shani, second profession BSMS. The second prize goes to Mr. Namashivayan, second profession BSMS, junior batch. And the third prize goes to Miss Agalya, second profession. Thank you, dignitaries. Congratulations to all the prize winners for their.
I saw people clapping, for a few people well and a few people little. So my request is after I finish, clap nicely so that I go happy from here. Because I love Siddha students for one thing, that they are so obedient. I told Martha sir in the morning that the drop of the hat, we call Siddha people to India Medical University. We are really happy because they really behave themselves. They listen to the CME. கேட்டு முடிச்சிட்டு இந்த ஓடி நடுவுல போய் இந்த ஒரு ரெஸ்ட்லெஸ் ஆகி அதெல்லாம் எதுவும் பண்ணாதுங்க. So we really love you all. So very sincere. முன்னாடியே சார் சொல்லிட்டேன். சரி, நான் actually சாரை பிடிச்சி வச்சிருக்கேன், டைரக்டர் சாரை, because he told about globalization, which is extremely important. இப்போ வந்து hot cake மாதிரி. ஏன்னா சென்ட்ரல் கவர்மெண்ட் மோடி ஜி is extremely attached to Ayush, and so please make the best out of it. So one thing is, in the airport poninganalum sir, there is a Siddha outlet. Is it in India? I'm not sure. Outside India, everywhere there is a small outlet. So Siddha medicine definitely is known outside. The problem with us is, if they tell us, we like it. If we tell each other, we don't like it. That is one thing. And the second thing is, I request all of you to think of small things. ஒரு small, இப்போ sir சொன்னது வந்து குடிநீர், கபசுர குடிநீர் during COVID, Christian sir was talking. So அதுல வந்து there are so many components which are making that குடிநீர். It's not one single drug. இல்லையா? அது தெரியும் இல்லையா? So அந்த combinationல எந்த drug most useful, எந்த drug antiviral இருக்கு அப்படிங்கறத, if you can come and do an NMR study. What is NMR? Any guess? No? Okay, so I think you all should look because you all have cell phones with you. Takarnu paathu sollirlame. Right. So NMR studies, you can do so many to prove yourself, and unless it is proved, science is very difficult to take it down. So sir sonna madri globalization of Siddha medicine, this is the time. And Tamil Nadu is the best place because we started the Siddha medicine. So ingerna the ripples vandu romba fast ah pogum. So it is a request from all of you, from sir and the rest of the team, to please start off research somewhere, and all of us are here to help you, whichever way we can. Thank you, sir. Now, I thank the organizing secretary, usual talk vandalam. Dr. Marga sir.
Professor at the Tamil Nadu Dr. MGR Medical University, has a career spanning over 25 years, encompassing both academic and administrative responsibilities. She earned her MBBS degree in 1990, followed by an MD in microbiology from Madras University in 1998. In 2023, she completed a PhD in medical microbiology from the same university, further deepening her expertise in the field. She began her career as a tutor in microbiology and has since held positions including assistant professor, associate professor, and professor. Her administrative roles have included head of the department, dean, registrar, and chairperson of the board of studies. She is a member of various councils and professional bodies such as the Entrepreneurship Development Institute and the Indian Council of Medical Research, and has been involved in academic collaborations with institutions including ICMR, NIV, and the Tanuvas. Dr. Pushkala has presented at international conferences in the US, UK, and Singapore. She has served as a speaker at several national level CME sessions and seminars held in institutions such as the Tamil Nadu Dr. MGR Medical University, Loyola College, and Yoshimi University, Japan, covering a wide range of topics. She has authored and co-authored over 30 publications in journals such as the Asian Journal of Transfusion Medicine, Journal of Biomedical Sciences, and Journal of Clinical and Cellular Immunology, focusing on subjects ranging from blood safety and immunology to diabetes, hepatitis, COVID-19 research, and molecular docking studies. Her publications are complemented by 2 authored books, Basic and Advanced Life Support, Good Clinical and Laboratory Practices, and Homeo Vision. She has also led or participated in several research projects, including zero surveillance of HPV, the One Health Consortium, and COVID screening, supported by funding agencies such as UGC, TBT, and ICMR. Dr. Pushkala has received several notable awards, including the Best Thesis Award from Madras University in 2024, the COVID Warrior Award from the Health Minister in 2021, and the Best Doctor Award from the Tamil Nadu Dr. MGR Medical University in 2009. National Institute of Siddha takes immense pride and pleasure in welcoming you, Doctor.
Thank you so much. Nice. Thank you, Meera ma'am. Right. So I thank, thank you all of you. So I thank Dr. Madhu. See, one short note about Sir. Sir Balakrishnan Sir or the students from Madurai, they have gone up to Fulbright scholarship. So the Pataponga, you tap him, you will get lot of resources. Sir, in your Kanambita. So, he has students in Israel, in Fulbright, Ramsamy Award, all his students have come. So, all of you, please take a note. If you just tap him at the right moment and you get a proposal done, you get a grant which he is ready to again, mentor you for it. You can reach any heights. Believe me, I have seen his students because they have worked with me also. Adala, please take good note of Dr. Mardu. And as Christian sir already told, yes, we were associated. He makes me feel quite old, but then fine. Thank you so much for acknowledging this thing. I know the rest of them. As sir said, I was then the registrar when sir was there in transfusion. Sir came to us for transfusion medicine. So yes, we've had an association with NIS for a very, very long period. I thank every one of you to have invited me. A special thanks to Meera madam. In a usual kalayil in the WhatsApp edunchonna good morning edo or flower or Sami padam I had a very nice thing today. hearty welcome to NIS. யாருமே நம்ம காலங்காலத்தால 5:30 வெல்கம்லாம் சொன்னதே கிடையாது. It was from Veena Madam today. Thank you so much, ma'am, for that encouraging, good morning thought. Dr. Saravanan coincidentally, is or was my examiner for this PhD which we did, and that was in hepatitis. So you know how big he is in hepatitis now, so he'll be after me talking about hepatitis, which I don't know if I can manage, or they are anyway videoing it, sir. So I'll take the recording and listen to it later. Right, so coming back, let's start with our topic. இது ஒரு வித்தியாசமான ஸ்னீக் பீக்லாம் எழுதியிருக்கேன், பட் அது வந்து ஜஸ்ட் இங்கிலீஷ்காக தான். More so, immunological techniques அப்படின்னா என்ன கண்டுபிடிக்க போறோம்? சார் சொன்ன மாதிரி we are going to prevent something, இல்ல வந்த ஸ்டேஜுக்கு முன்னாடியே கண்டுபிடிக்கிறதா, இல்ல விண்டோ பீரியட்னா என்னன்னு தெரியுமா? Yes or no? Okay, so when the infection is in, ஆனா அதை டிடெக்ட் பண்ண முடியாது, in that stage is called the window period, which is very dangerous. Yeah, the disease is in already, but you're not able to detect it, which means you don't even know that you have the disease. You are happily giving it to everyone also. That is called the window period. So we won't dwell into that now because that's not my topic. Now, on the techniques, why are we bothered about this immunology? நம்மளோட பாடியில என்னெல்லாம் first mechanisms இருக்கு? What is this immune system we are talking about in our body which is trying to fight against the foreign particle? Am I clear? You can raise your hands in case you have any problem, because it will be a mixture of both Tamil and English. I hope you are comfortable with it. Are you? Yes, ma'am. Good. And the 30 வருஷமா teaching line la irundadanaல I like to ask questions. So I don't like a dialogue. So unfortunately or fortunately, you must come with me, because I can point out to somebody from here, stage konja higher irundadanaல I can tell, yes, that person, this person அப்படினு. So you have to come with me and just say, wherever you have an issue, doesn't matter. You don't need to know everything. Even I don't know. So we can always sort it out that way. Otherwise, it's never going to be an interesting thing. இது வந்து இந்த டயலாக் நான் மட்டும் பேசிட்டு இருந்தேன்னா என் மூஞ்சே இன்னும் 1 மணி நேரம் பார்க்கணும் நீங்களாம். And then it's very difficult. So I will be starting, but I want complete attention because we've really spent our time here. Right, so now we go to the immune system. I hope it changes here. What is this immune system? Right from the head up to the toes, we have lot of immunological systems in place. முதல்ல இந்த lymphatic system. Lymphatic system na என்னது? Any one of you? நெறி கட்டுறதுன்னு கேட்டிருக்கீங்களா? புண்ணு வந்தோன்னு நெறி கட்டும்னு கேட்டிருக்கீங்களா? Yes madam. கேட்டிருக்கோமா நம்ம? Yes or no? No? புண்ணு வந்தது, கால்ல புண்ணு வந்தது நெறி கட்டுமா? Yes ma'am. Okay. So yes ma'am. அதனால the lymphatic system is an important part, right? So from there, if we start, almost all the organs, liver, spleen, blood, everything has cells which can help us with the immune system. Okay? So that is why it is so important to know what are the cells which are responsible for it. நம்ம என்ன இந்த டெக்னிக்ல கண்டுபிடிக்க போறோம், என்ன செல்ஸ் பார்க்க போறோம். So next is the immune response. So sir சொன்ன மாதிரி it is free from burden. Immunity, immunology, immune system, immune response, எல்லாமே I am away from everything. I am free from all kinds of diseases. So that is what is immunity. So adla you have 3 types. One is the first line. First line is skin. So say a mosquito bites you. What happens to it? Can one of you tell me? Excellent. So there is something called a wheal and flare reaction or an inflammation. So on the area was orange and the red dye with it. So what happens? All the neutrophils come running to push whatever has bitten us. The little bit of, extra material which has been sunk into the skin wants to be thrown out. So that is the first thing. So other one, the skin has been provided by the Supremo, whatever, nature, God, whatever you call it. So that is the first line. And the mucous membrane, adho first line da. Mucous membrane membrane na enna? Ipo vandi ஓட்டிட்டு இருக்கும்போது ஏதோ ஒரு கண்ணுல வந்து விழுறது. What happens immediately? Watering. Good. So immediately there is some water. We blink our eyes. The water flows that dirty thing out. So the secretions of mucous membrane. So that is also the first line. The next is second line defense. Second line defense is phagocytosis. Abdina, nammaloda few cells which I will be describing soon will go catch hold of the antigen, take it inside, completely kill it. Okay, so that is called the phagocytosis and then the cell lysis. So we'll go into that a little later. The third line, of course, is the lymphocytes or the antibodies and the rest of it. Now we come on to the cells of the immune system, which I already told you. என்ன செல்ஸ் இப்போ நம்ம சொன்னோம் இல்லையா, ஸ்கின்ல வந்து இம்மீடியட்டா ரெட் ஆயிடுச்சு அப்படின்னு. ஸோ இந்த ரெட் ஆனது எதுனால? நியூட்ரோஃபில்ஸ்னால. ஸோ தேர் ஆர் டூ டைப்ஸ் ஆஃப் செல்ஸ் விச் ஆர் ரெஸ்பான்சிபிள் ஃபார் தி என்டையர் இம்யூன் சிஸ்டம். இப்போ என்னெல்லாம் செல்ஸ்? மேபி த லாஸ்ட் பர்சன் கேன் டெல் மீ தேர், த லேடி தேர். எஸ் மேடம், வாட் ஆர் த டூ செல்ஸ் விச் வி ஆல்வேஸ் டாக் ஆஃப்? Yes, very good. Someone here answered. B cell and T cell. It's on the slide itself. So, B cell and T cell. B, abdina, it can be bursts of lumbricus or bone marrow. Innona, T cells. T cell is otherwise the thymus. Excellent. So, you have two types of cell which causes all this kind of confusion. So, number, ipo, idunga rendu enna vela seyyirudhu அப்படிங்கறத தான் நம்ம டெக்னிக்ஸ்ல கண்டுபிடிக்க போறோம். And that is why we are laying so much of importance on the B cells. இந்த B cells வந்து pre antigen, post antigen, full lineage கொடுத்திருக்கேன். But more இதெல்லாம் இந்த theoretical விட B cells mature in bone marrow, T cells matures in thymus. அதனால தான் T. Okay, so these are the two major cells that are responsible for the immune system. Now we go on to the next one. Okay, so the same thing I told you. Now T cells எடுத்துட்டோம்னா it produces variety of cells. We'll come to that a little later. But in the T cells வந்து is generally the one which presents the antigen. இப்போ ஏதோ ஒரு foreign material வருதுன்னா அது நம்ம பாடிக்கு அதை present பண்ணணும். அது சொல்லணும், இது வந்து ஒரு thief வருது, இது வெளியிலேருந்து ஒரு விஷயம் உள்ள வருது. ஸோ அந்த பிரசன்டேஷனுக்கு T cell is very very important. But when it comes to B cells, they are the ones who act against this foreign material. Is this clear? So we are going to find out what is this T cell, what is this B cell. Now T cell வந்து பாடியில இருக்கா இல்லையா அப்படின்னு கண்டுபிடிக்கிறதுக்கு easiest thing is sheep cell rosette formation. Rosette அப்படினா பியூட்டிஃபுல்லா அந்த ரெட் செல்ஸ் எல்லாம் T cells சுத்தி நிக்கும், which means T cells are present in that serum. So what is that called? Rosette formation. Okay, so that rosette form ஆச்சு அப்படினாலே T cells இருக்குன்னு அர்த்தம், right? B cells தான் இனிமே நம்ம ஃபுல்லா பேச போறோம், because T cells are important to one extent. After that, it is only the B cells which we are trying to detect by the techniques that we are doing. If you have any questions, I think it's better you address it to me than talk amongst each other, because it will become difficult then. So the next part of it is now the B cells. B cell is usually found B cell is found in the plasma, right, or the serum, in the fluid part. But it has two huge things. One வந்து B cell differentiates as plasma cells or memory cells. Sorry, it differentiates as memory cells, and second as plasma cells. Plasma cell தான் antibodies. இது கேள்விப்பட்டிருக்கோமா நம்ம? Yes ma'am. Good. So antibodies produce பண்றது வந்து B cell, and that is found in serum. It is in the fluid state. Why are we insisting? Because techniques for finding out the B cell is only by taking the serum or the plasma. Is this clear? It's an extracellular. It's வெளியில செல்லுல இருந்து வெளியில float ஆயிட்டு இருக்கும். Okay, so that is antibody. இப்போ நான் சொன்னோம் இல்லையா, memory cell and antibody cell சொன்னோம். இந்த memory cellல என்ன ஒரு advantage? Any guess? Any one of you? Excellent. So she is talking about secondary response. Any daily use, I come to that, madam, but can we talk about any daily use of this? I mean, use of in scientifically because of this memory cell? Excellent. So somebody said about this vaccine. Very, very nice. So vaccine la enna pandrom? First ஒரு injection கொடுக்கிறோம். அந்த injection is say hepatitis A ஆரம்பிப்போம் நம்ம, because you are going to listen to that. அதுக்கு வந்து we give them a small dose of hepatitis, killed virus, inactivated virus. Now we have synthetic, everything is available, safe ones. கொடுத்தோம் என்ன பண்ணுது? T cell அதை வாங்கி B cell க்கு present பண்ணி B cell will produce memory and plasma cells. Okay, because it is not high enough, it does not produce any kind of inflammation and try to throw it out. அதெல்லாம் பண்ணாது. Instead it will produce memory. It will cause, oh, இவன் வந்திருக்கான் உள்ள ஏற்கனவே அப்படின்னு. So next time வரும்போது wonderful react பண்ண முடியும். Immediately you've come before, no, now I have a battalion ready with me. I throw it at you so that you are out of the system. So that is what is the advantage of B cell. Is that clear, ma'am? Okay. So the next now is, as I said, humoral, where are we? I don't even know. Okay, T lymphocytes. I think we have crossed this. I'll just tell one example of this. T cell is cell mediated immunity, which means B cell is found in serum, T cell is found in the cells, and what is the test which I told you? Rosette. Excellent. So rosette formation is the one. These are all extremely basic. Now don't ask me whether we are doing it now. We still do it, but quite rarely. Only if you understand these things can we build on the rest of it. So now T cells, there is something called major T4 and T8 cells. And what do we call this? CD4, CD8. CD4 no enna or bell adikida? Enna adikide? Cluster of differentiation. Excellent. CD or the full form. CD4 edhula paapom? HIV. Yes. So CD4 is one of the most important marker. Initial days was CD4. And how do we see or determine the CD4? Usually by flow cytometer. Okay. So that we will talk of it also. So CD4 வந்து helper cell. Helper cell na என்னது? Antigen வாங்கி என்ன சொன்னோம்? எங்க present பண்ணும் அது? B cell க்கு present பண்ணும். Excellent. So B cell க்கு present பண்ணும்போது இந்த helper cells பண்ணிட்டு அங்க antibodies எல்லாம் produce பண்ண ஆரம்பிச்சிடும். So this is called helper T cell or CD4. Is this clear? Yeah. Now help பண்ணிட்டே இருந்து எக்கச்சக்கமா immunity பண்ணி அது வந்து என்ன ஆகும்? எப்பவுமே even if it is அமிர்தம், if it is more, it's going to be poisonous. இல்லையா? So அதே மாதிரி antibody எக்கச்சக்கமா வந்துருச்சுன்னா what will it lead to? It may lead to cancer. So எங்கேயோ ஒரு இடத்துல நிறுத்தணும். So that is called suppressive cell or the T8 cell. Is this clear? So we have two major things T cell la. Enna? T4 and T8. Excellent. So T4 and T8. Okay? Clear? Right. So now B cell causes, now I've gone ahead with this. So I've told you about humoral immunity. Where do we check with humoral immunity? What is responsible? Which cell is responsible for humoral immunity? B cells. I'm so happy. Good. So the next one is cell mediated. Either responsible? T cells. Excellent. So we have done all this. Now you are clear up till here? Good. Now we go on to something called the immune system. With the cell எல்லாம் பார்த்தாச்சு, எங்க இது வருதுன்னு பார்த்தாச்சு, two different cells பார்த்தாச்சு, how does it come and produce பார்த்தாச்சு. How do they mature? Where do they mature? All this we have seen. Now there's something called two systems. Innate, adaptive. Innate அப்படின்னா என்னது? Excellent. ஏற்கனவே இருக்கிறது innate. Again, நான் சொன்ன மாதிரி it's a supremo which is already given this innate immunity. So the innate immunity and the adaptive or the acquired immunity, this is one of them. Now innately enna சொன்னோம் நம்ம first skin, mucous membrane, right? So secretions of mucous membrane is from an IgE. அது நம்ம என்னன்னு அப்புறம் பார்ப்போம், but just remember this. So this is on the mucous membrane or anywhere in the gut, wherever there is an irritation, this is the first thing that gets formed. So that is the antibody which is formed, that is innate of the mucous membrane. Right. Now acquired you have two types. You have an active and a passive. Now active அப்படினாலே you get the disease. அப்போ ஒண்ணு active நம்ம ஏதோ ஒண்ணு we are trying to create and trying to push the disease out. So we are actively involved in it. Passive என்ன வரும்? என்ன வரும் passive? Vaccine. Recombinant இல்ல vaccine, any vaccine. We are trying to mimic the disease. Disease வந்தா நம்ம prepared இருக்கணும்ங்கிறதுக்காக அந்த vaccine கொடுக்கிறது. So that vaccine is going to be the active immunity one part. Now when we go on to passive, there are two things again. One is vertical. What do we mean by that? Excellent. Mother to child. So it's vertical. The second one is blood transfusion. Prepared ஒருத்தர் கொடுப்பாரு டோனர் டேந்து அவங்க இன்ஃபெக்டட் செல்ல நம்ம ஹாப்பியா வாங்கிப்போம், நம்ம இன்ஃபெக்ட் ஆயிடும். So that is passive. So this is what is the classification. Is this clear? Okay. Now we come to the real culprit of the whole day, antigen. That's the foreign material, right? So in the foreign material ku da nam ivlo nice sanda potutukom. We want to find out the techniques which can either find out the antigen itself or the reaction to the antigen. Reaction என்னவா இருக்கும்? Antibody production or cytokine production. அது நம்ம cytokine என்னன்னு ரெண்டு நிமிஷத்துல வரலாம். But T cell production. So any of this detection will detect that there is some response going on to this antigen, right? Yes? Okay. So now what is the most important part of an antigen called? See, antigens are huge. They are immunogenic. Well, not gone onto the slide. Okay. So they are immunogenic, antigen specificity. There are so many things involved, starting from molecular weight, what is the complexity, what is the foreignness. Because of the foreigner இருந்தா தான் நம்ம கண்டுபிடிக்க போறோம். நம்மளோடதா இருந்தா நம்ம அத போய் சண்டை போட போறோம். It is our person. So we are not going to fight against it. If it's a foreign thing, you're going to fight and say, get out from here. I don't want you. So that kind of characteristics of an antigen is determined by few sites on the antigen. Entire antigen தப்பு கிடையாது. And the sites க்கு பேர் என்னது? Any guess? Excellent. Epitope. Who told this? I told that. Good, I'm telling you. Somebody said this? Very good. So the same biochemistry person. Right. So this is epitope. Epitome is extremely important because that is the site which is antigenic. It is irritating the body system, right? So that is called the epitope. Now we come on to types of antigen. Exogenous, வெளியிலேந்து வருது. Endogenous, yes, and autoantigen. One word on autoantigen. But there are some antigens inside our body which இப்போ நம்ம இவ்வளவு நாள் என்ன சொல்லிட்டு இருந்தோம்? Antigen is a foreign material. Last benchல கண்ணாடி போட்டு வச்சிருக்காங்கல்ல, somebody is looking at the cell phone. Can you just tell me what is the? Yes, madam. Yes, you, madam. Can you just tell me what is the antigen which is already present inside our body? Any guess? It's a very common antibody. We do the detection of this, not antibody, antigen. We do the detection of this antigen at school entry, driving license. எதாவது ப்ரூவ் வேணுமா இன்னும்? என்ன பண்ணுவோம் டிரைவிங் லைசென்ஸ் வாங்கும் போது? யாரெல்லாம் வச்சிருக்கீங்க டிரைவிங் லைசென்ஸ்? வச்சிருக்கீங்களா சார்? என்ன வாங்குறீங்க நீங்க? என்னலாம் எழுதுறீங்க அதுல? Yes, somebody saying that. Blood grouping. மீரா மேடம் இல்ல, வேற எல்லாரும் சொல்லணும். Blood grouping, right? So A group, உங்க group என்ன மேடம்? உங்க group தெரியுமா தெரியாதா? O வேணாம். அப்ப we take something concrete. A or B. சரியா? இப்போ A groupல என்ன antigen இருக்கணும்? பாடியிலேயே இருந்தது அப்படின்னா why are we not fighting against it? We should have fought. No, it is an antigen. We are calling it an antigen which is on the surface of the red cells. So why should we not fight against it? It is something like a self antigen which we are born with. So the system gets tolerant. So that is called immune tolerance to that particular antigen. அதனால அது வந்து நம்மள இரிட்டேட் பண்ணாது. சீரம்ல என்ன இருக்கும்? ஏல antibody to B. So that also is there. That is called auto antibody, which again is not dangerous because it's there from birth. Right? We'll come to that a little later. Now, these are the characteristics. Sorry. Oh, I just want to tell you about this. There's something called a complete antigen. There's something called hapten, which is extremely important. In the half-baked knowledge which எவ்வளவு பேர் இந்த கூகுளை வச்சு எல்லா டாக்டர்ஸையும் படிச்சிட்டு இருக்காங்கல்ல, அந்த மாதிரி தான் இந்த ஹாப்டன்ஸ். அதை ஸ்டார்ட் பண்ணி விட்டுட்டு போயிடும் அந்த ரியாக்ஷனை. அது ஃபுல்லாவும் வராது, அது பாதியாவும் அபார்ட் ஆகாது. So that is the problem about incomplete antigen or haptens, because detection of this becomes extremely difficult. That is how we have gone on to talk about microarray, which I will be talking also. So அதெல்லாம் பண்றது இந்த half-baked haptensனால, incomplete antigensனால. They will start a reaction, but they don't know how to complete it because they're not full. Okay, so that is one part. Okay, now the best part of antigen is it can be anything. It can be a polysaccharide, it can be a sugar, it can be a protein, it can be any of these things. So, but an antibody on the other hand is only and only a protein which is produced by antibody is produced by B cells. So this is only a protein. This is the basic structure, Y-shaped, where you have the Fc portion which combines C, abdina, it combines to the complement. Yes, have you heard of complement? Okay, right. There is a variable region and there is a permanent or a fixed region. Now, ஏன் இதெல்லாம் சொல்றோம்? இந்த ஆன்டிபாடி ஆன்டிஜனோட போய் கம்பைன் ஆகும். So in the principle of combination between the antigen and the antibody is the basis of all the techniques. So you should know this part extremely well. So antigen la epitope is very important. Antibody la this Fc portion, the variable portion and the combined portion. All the three are very important. Now we come on to this. I think I have already mentioned. This is almost 20% and it is found in the red cells or plasma. Plasma. Remember that. So that is why we think of serum or the fluid part of the blood to be taken for testing. We don't take the cells. Cells we do only for T cells. And then of course for HLA, that also I will be saying, but that will be all later. These are all smaller things. Major வந்து நம்ம antibody detection or antigen detection in the serum only is what we do. Right. Now these are the types of antibodies. First antigen வந்தோடனா IgD is the one that's produced. IgD ல இருந்து IgM. IgM is a pentamer. Pentana என்னது? 5. So 5 molecules, which means 10 places. ஏன்னா Y சொன்னேன் இல்லையா? So 5ல 10 antigens சேரலாம், which means such a huge matrix it will form. So that is what we mean by IgM, which is big. So IgD cannot be detected. IgD க்கு அடுத்ததே IgM. Is this part clear? So IgM first produce ஆகுமா? Yes. So the last person again, the boy there. Yes, sir. IgM is what valency, sir? Pentamer, which means it is 5 immunoglobulins together which can combine with how many antigens? 10 antigens. Excellent. Sit down, please. So 10 antigens which forms a huge big matrix which is easily detectable is called the IgM antibody. first produce are the IgM. So somebody talked of primary response, secondary response. So adukku namme ipo varuvom. IgG is the second one. IgG is only a monomer or a dimer. Sorry, it's a dimer. So it's a small one only, but it is produced as a second antibody. Okay, so first one is IgD, which cannot be detected. So adha pathi nam usually pesa maatom. We don't have any techniques to detect the IgD at all because it gets converted very fast into IgM. And the IgM after some time becomes the next molecule that comes in is IgG. Right? So these two are the ones which detect through and through for diseases. It's either IgM or IgG that we detect. IgE enga varum sonnoma? Mucosal secretions, okay, allergic reactions from histamines, from mast cells. They are the ones which produce the IgE. If Ig is high, it means you are allergic to something. So that is what you check when you have eosinophilia, when there is a skin rash. Appalam kekkumbodhu they talk about IgE. You know, the IgE is meant for anaphylaxis, any kind of allergic reaction. Right, then we have one more and that is called the IgA. IgA enga produce agum? Any guess? Colostrum. Very good. So IgA is in the colostrum, which is extremely important. We won't go into the topic. I think it's a full continuous CME which we did on colostrum as well. So IgA is again secreted in the secretions of the body, which is very useful. So now we go on to the next one where I want to fix a few things. IgM is the one we detect for blood group. Okay, wow. Ipo antibody to that A antigen sonolya. How to detect group A? Abdina anti-A is an IgM. It reacts only at 24 degrees. Abdina நம்ம blood group எங்க பண்ணுவோம் நம்ம? Where do we do the IgM or blood grouping? Yes, madam. எங்க பண்ணுவோம்? Room temperature 24, where the whole place is AC. We expect the lab to be AC, and we do the blood grouping at 24 degrees. Why is it important? I am saying because IgM reacts at 24 degrees. இப்போ புரியுதா why we are insisting on such small things? ஏன் நம்ம ரூம் டெம்பரேச்சர் பண்ணா எல்லா ஏசி ஆஃப் பண்ணிட்டு வேர்த்து பண்றது is not the way to do it. அப்படினா IgM வேலை செய்யாது. So we do it at 24 degrees, whereas IgG is done at 37 degrees. IgG blood group ஓட ரிலேட்டடா என்ன இருக்கும்? Yes madam, blue chudidar ma'am. Yes. You. Yeah, you only. நீங்க தான். நீங்க இல்ல மேடம். Yeah, not you. Yeah, tell me. என்ன क्वेश्चन தெரியல ஏனா I know that. IgG blood grouping சம்பந்தப்பட்டதுல என்ன பாப்போம்? எங்க பாப்போம் IgG? Don't know? Any guess? RH, excellent. DU. So RH is the IgG. So RH IgG na it is at 37 degrees. ஏன் நம்ம சொல்றோம் திருப்பி? IgG reacts only at 37 degrees. அதனால DU test போடும்போது அது இன்குபேட்டர்ல தூக்கி தான் வைக்கணும். அது ரூம் டெம்பரேச்சர்ல பண்ண கூடாது. You will have to keep it inside the incubator, which is set at 37 degrees. Now do you understand why? Yes? Okay. So now the next thing about IgG is it crosses the placenta. இதனால என்ன? Have we read about children who are positive and mothers who are negative? Yes. So on the time of delivery, the IgG which is released from the child or the fetus will go into the mother and cause lot of antibodies inside the mother. So the next kid varumbodhu the mother will fight it against the kid. So that is why we have anti-D injection. So again that's a different topic. But why are we invoking that now? It is because it crosses the placenta. அப்போ இன்னும் ஒரே ஒரு இது IG ஒரு பாப்பாக்கு HIV பாசிட்டிவ்னு வருது or HBV பாசிட்டிவ்னு வருது. Right? And the papala IgG paakaram. What does that signify? Is it from the mother? Yes. Vertical transmission crosses the placenta. So, if the child born is positive for some disease with IgG, then it is from the mother. Vertical transmission, which means you can still wait happily. The child may react and throw the virus out. It's a good sign. Okay, but if they have IgM, it means IgM irundha enna vannum? The child has acquired it. The child has got it from someone. So that we have to treat separately. Vertical transmission is treated separately. Is this part clear? Okay. So that is why we are insisting on activity or characteristics of each antibody. That is how we can interpret the test result. But test எல்லாம் பண்ணி முடிச்சிட்டு எப்படி interpret பண்ணனும்னே தெரியாது. What are you going to tell the clinician ங்கறது we have to know. Or what does he expect from us ங்கறது தெரியணும். So that is why we are trying to dive into all this part. Is this part okay? Right. Now I don't know where we are. I think. Okay. So yeah. Now about the primary response. So the first response is called the IgM, and the second response. So first response takes some time. Why? Because the T cell has to capture the antigen, show it to the immune system or the B cells, and then it produces the antibodies which tries to throw out the antigen. So this entire thing is called the primary response, which comes from IgM. Second time varumbodhu already formed, already memory cells la irukku. So another secondary response by IgG is very fast. So primary response is slow. Secondary response by IgG is very fast. So if for IgM kandupidichom or disease, say we talk of HIV IgM. I don't think HIV venda. We'll talk of leptospira. Al IgM kandupidicha enna meaning? Acquired at that time. He is acute fellow. He has the infection actively inside. He has just started developing the antibodies. So he is dangerous guy. He is a carrier, he is dangerous, he may give it to anybody he wants. So that is the problem about IgM present in serum. Okay, if it is IgG, yes, so the person has gone through the acute phase and come to the secondary phase. So he still is a little safe. It's not that he is completely out of the disease, but at least he won't be transmitting as fast as an acute case. He may be a chronic case. Now again, that is again a separate chapter about carriers, super carriers. Maybe he'll talk of it. But then that is how it goes as IgG. This is the importance of IgG and IgM detection. Is this part very clear? Okay. So now we are worried about the interaction between the antigen and the antibody only. And that is the base of all our techniques, which I already told you. Either there is something called self-antigen blood grouping. I have gone through all this. Any disease which is produced because of an antibody against our own good cells. Excellent. Autoimmune diseases. Anyone, any name of that? One of you? Rheumatoid arthritis. It is against HLA? D23? Okay. It is fine. It is against one part of our chromosome itself, which is a part of our cell. Okay. So that is why it is called an autoimmune disorder. It is against our own immunity. Okay. Now, this is the interaction. Interaction between antigen antibody is only a physical reaction. All these words have to be noted, because these are the words which are very important where we may go wrong with the technique. It is only a physical reaction. See, chemical reaction, chemical reaction, sodium chloride form ஆயிடுச்சு, sodium and chloride, sodium chloride form ஆயிடுச்சுனா உடைக்கவே முடியாது. Whereas antigen antibody reaction ஆனா, if you vibrate it violently, it will break, because it is just a physical reaction. Okay, it's just the hydrogen bonds. Yes, the person last part of it. Can you just tell me, madam? Yes. How is the antigen, which part of the antigen is being presented? Okay. So we have the epitope of the antigen which is being presented. And see, this is the diagram, sorry, which gets activated. Sit down, please. Just try and listen to it. That's all. We're spending all our energy on it. Okay? So this is antigen presenting cell. That is a T lymphocyte. Did we hear that? Yes. So the epitope of the antigen presenting cells, it shows it to the B cell. B cell gets mature, antibody gets formed. So that is the diagram there on top. Then from there it can be opsonization, membrane attack complex, it releases the complement, finally causes, somebody said in the first part, inflammation. And the skin la nadakra oru kutti red varudhu pathingala, mosquito bite, ant bite nalla, it inflow process nadandu and the neutrophils odi varum and edathukku. So this is the process that is taking place inside the body by the time inflammation takes place. Okay, so now we should know what are the factors which affect the antigen antibody. Temperature. Enga nama temperature patti pesinum? The third person here. Yeah. Where did we talk of temperature, madam? Excellent. So IgM we said is at 24, IgG is at 37. So temperature is extremely important. Then pH, of course, the incubation time, or time கொடுக்கணும் இல்லையா, it's only a physical reaction. So அவங்க ரெண்டு பேருக்கும் கொஞ்சம் time கொடுத்து அவங்க complex form பண்றாங்களான்னு பார்க்கணும். Centrifugation, as I told you, antigens and antibody is a physical reaction. So you have to be very careful about it. All this fine. I have a small thing in this where you have something called the prozone phenomenon. அப்படின்னா antigen எவ்வளவு இருக்கு, antibody எவ்வளவு இருக்கு. So ஒரு ஆன்டிபாடியில 10 ஆன்டிஜன் சேரலாம் சொன்னோம் IgMல. So அத்தனை ஆன்டிபாடி இருந்து நிறைய ஆன்டிஜன்ஸ் இருந்தா the correct complex will be formed. If antigens are more, the antibody will not be able to capture all the antigens. So we will lose sight. So we'll have to do any guess? சீரம்ல நிறைய ஆன்டிஜன் இருக்கு. We are trying to detect it. நிறைய ஆன்டிஜன் இருக்கிறதுனால என்ன பண்ணணும் நம்ம? Pardon? Dilution. You dilute that serum. Either it is called serial dilution or doubling dilution. Again, I won't go into that. Biochemistry ma'am can do that. But that has to be done. Serial dilution, serial or doubling or pannatha and the antigen amount குறையும். குறைஞ்சாதான் இது கரெக்டா காம்ப்ளெக்ஸ் ஃபார்ம் ஆகும். So that is a very, very important constraint problem in the technique. If the antigen is high, we just cannot say, edhum varalappa, adha negative. No, huge positive is being given as negative, where we may need to do a dilution. Clear? Okay. So now, antigen antibody, this I have finished, prozone phenomenon. Now we go on to lock and key. As I said, it's just a non-covalent bond, which is just a hydrogen bond between the antigen and the antibody. It is very much reversible. Okay? Are you all with me now? Till here? Okay. Now we told about this primary. We told about the subsequent. There is something called the isoantibodies. Did we talk of that? Why? Autoantibodies is otherwise called isoantibodies. Avladhan. Just a name given to it. That somebody told us about rheumatoid arthritis. That is an isoantibody. It is an autoantibody. Okay. But we, of course, in called a prozone, there is something called an antigen-antibody complex, and that is all, enhanced by complement. So there is something called a complement fixation test. So adum oru technique. So antigen antibody in the reaction nadakradhukku we have something called a complement which comes in to push the reaction forward. It's something like an enzyme which pushes that whole reaction to take place, and that is called the complement fixation reaction or test, CFT. which is done for many tests. Again, I'll be enumerating a few minutes later. But this is one of the techniques which we use. So now you all know, we find out the T cells by just rosette. B cells, of course, we have flow cytometer for determining the CD4 and CD8. Did I say that? Yeah. So, flow cytometer, we have rosette cell, we have many other things like colony stimulating factor, macrophage stimulating factor. All these are the ones released by T cells. So, idallamum namba vandu ELISA la kandupidikkalaam. So, that is T cell. B cell ku varumbodhu we have antibody detection, we have complement detection, we have different types of antibodies which can be detected. Is this part clear? So now we are going to the techniques. Right. Now there are 4 types of major. What is written there? Okay. Now I'll just move on with this. All this I have finished. This is the complement system which I spoke of. You have a classic pathway and a alternate pathway. And it's slightly right? Okay. So now there are 4 types of reactions which we are trying to bring into the techniques. So the antigen-antibody reaction da nama kandupidikkanum. The antigen-antibody reactions are 4 types. One is agglutination, precipitation, complement fixation, neutralization. That is all. These 4 things, if you all know what is the basis of all this, this will form the basis of all the tests that we are doing in immunology. Now, what happens here? My stalwarts are back up. It's not going forward. Can somebody help me? Where is my Krishna? Yes, come. Okay, so we go on. Next slide. Yeah. So, as I said, there are the 4 reactions which we talk of. Yeah, I can. Thank you. Agglutination. What is the next one? Precipitation. Excellent. Third? Complement fixation. And the 4th? Excellent. So I won't go into the details of this, but just to tell you that neutralization, agglutination, precipitation all leads to phagocytosis. Did we talk of this? Yes. What was the cell which was responsible for the phagocytosis? Macrophage. And how do we detect the macrophage? Okay. So we have macrophage colony stimulating factor. Did I speak of this? Colony stimulating factors? Yeah. Oh, so there's a brief time of. Okay, let's not go into it. There is something called a colony stimulating factor. I told you about the T cells, where you can go through flow cytometry or can go through all these factors. Are you now able to recollect? So colony stimulating factor is a very important one. Macrophage detection is a very important one, where you can check whether the whole thing has been phagocytized. Once it is, it gets lysed and thrown out. That's the last stage. So then the complement is one will also cause all this with the cell lysis. So that is the importance of complement in this. Clear? Yes or no? Very meek, yes. Why? Are you all with me? Yes, ma'am. Nice, ma'am. Yes, ma'am. Right. So now we go on to Ig. What is this blood grouping? What is the antibody which we are detecting in the group? See, anti-A which form? In the antibody detect pandrom? IgM. Remember this. It is IgM on the cells which is against the antigen which is already present on the red cell. So if a person is A, he has A antigen and we add IgM anti-A to it. How do we produce IgM against our own antigen? We inject that antigen into one of the animals, either a mouse or a rabbit, and you get the anti-human globulin. Is this part clear? We are injecting something into the animal. For that is a foreign material. So it's going to produce antibodies. That is the antibody which we use. Of course, now we have anti-human itself, right? But initially we used to do it only with a sheep or a rabbit. Okay. Now we come on to what is, now here this reaction is called the agglutination reaction. This is on this ends. What do we see in the serum? Same A la enna paapom serum la? Anti-B, not anti-A. Anti-A is the cell odinju pidum. So it is the anti-B which we are seeing in the serum. So adhe eppadi kandupidipom? There is something called the reverse blood grouping. Forward blood grouping is where we do the antigen detection. Reverse blood grouping is what we do about the antibodies in the serum. Is this part clear? We won't go into the significance of forward, reverse, after the blood group class I know. So we will now stop at this. The next part is flocculation. This is a part of agglutination. Right? So flocculation is, it forms floccules, fluffy materials. What is this test where it is positive or we see only as floccules? Again, age-old one. It is called VDRL. It is for the detection of syphilis. Very good. So you all know VDRL is a detection tool for syphilis. And what is the technique used? Flocculation. So this is the techniques. So first agglutination we used agglutination as a technique for blood group determination. When it comes to flocculation, we are using flocculation technique for VDRL. அது floccules இல்லையான்னு பயமா இருக்கு பார்த்தா தெரியல serial dilution போடணுமா? Why are we doing serial dilution? Because of Proson phenomenon. Excellent. So antigen excess இருக்கலாம், antibody excess இருக்கலாம். So அந்த இதை strip off பண்றதுக்காக தான் we do the dilution. So the dilution பண்ணி we are still not sure. ஐயோ இது பாசிட்டிவா நெகட்டிவா இது மைக்ரோஸ்கோப் கீழ பார்க்கணுமே அது floccules தெரியுதா? அதனால they have now said carbon particles add பண்ணிட்டாங்க, RPR add பண்ணிட்டாங்க. அழகா floccules form பண்ணனும், அந்த இடத்துல concentrated carbon particle வந்துரும். So macroscopica கூட you can see whether it is positive or not. Right? Clear? That is the second part. Somebody is very happy behind, I like it. Precipitation. So the precipitation is where the antigen meets the antibody. அது ரெண்டும் சேர்ந்த உடனே there will be a line formed. They precipitate because of the antigen-antibody reaction. As I told you, that is the base. If we have understood that part, இவ்வளவு நாள் சொன்னது உங்களுக்கு புரிஞ்சிச்சு அப்படின்னா அதுதான் base for all the techniques. Okay, so precipitation la vandu antigen will be coming or antibody will be coming from top, the other one will be coming from bottom. As soon as they meet, they form a precipitation line. So we finished agglutination, a part of it is flocculation, the third one now is precipitation. Right? Now this precipitation la most important thing, this is the precipitation line. Now the most important thing in this which we do is radial immunodiffusion, where we have the antibody or the antigen laid in the gel. Right? It is in the petri dish, full lay pannirvom. We cut wells and add the serum. Serum la enna irukum? Antibody irukum. So the antigen will be in the plate, antibody will come wherever they come at the optimum level. So that's a nice word. Optimum level between antigen and antibody vandona, that the precipitation line varum. Right? So it can be now used for various purposes. There are various techniques, so I won't go into all these details because it's so well given in your ordinary textbook like Panikar. Read that, it's wonderfully given. So I'm not going to go into these details, but I hope now you understand why are we talking of precipitation. Most important use of precipitation here is immunofluorescence. Right? Now what is this immunofluorescence? Fluorescing when the antibody is tagged or antigen is tagged with the fluorescence. Usually use it for Ben Jones proteins. Because that I've heard Siddha people talk a lot about that. Any idea of where we get the Bence-Jones proteins in urine? Okay. It comes in multiple myeloma. What is multiple myeloma? Is it cancer or normal? Cancer. So it is, multiple myeloma is a cancerous disease. I won't go again into those details, but those proteins can be found by the immunofluorescence, where you tag against the protein an antibody, you tag that antibody, it will go beautifully form a complex that is seen under fluorescence. In the microscope, fluorescent microscope. Excellent. So that part of it is clear. We see those bright, green color, fluorescent color, we know there is something wrong inside. That is the best way till date to find out so many of the big proteins. Immunofluorescence technique is just a tagging of the antibody towards the antigen or antigen towards the antibody. Is this clear? Okay. So we've come across 4 or 5 techniques already. So this is again just the principle of it, a diagrammatic representation of what I tried telling you. Now what? The next part of it is ELISA. Who's going to tell me the full form of ELISA? That's a nice chorus. So you all know what is ELISA. What is the enzyme which is commonly used? Any one person? One person? Absolute silence, Aisha. One enzyme? Yes. Again, I think it's Meera Madam telling, but doesn't matter. It is the HRP. What is the full form of HRP? Horseradish peroxidase. ACE அப்படினு வந்தாலே அது enzyme. அப்ப அதுக்கு substrate Meera Madam? Yes. Okay, so I think all that you guys have to read as homework. Otherwise, everyone is looking at each other's face and giggling away. I really don't know why. Sorry. What is the plate that we use for ELISA? Microtiter plate. Appadina, avlodha volume add pandrom. How many wells are there? 96. Some other guess? Good. So that is the ELISA plate. Now, something is bound to the surface of the plate. It can be an antigen or an antibody which is bound to the polystyrene of the ELISA plate. We can create it as in-house or we can get it from the manufacturer. Usually, we manufacture it வாங்கிட்டா அவங்க என்ன சொல்றாங்களோ verbatim அப்படினா word to word அவங்க என்ன சொல்றாங்களோ அதை follow பண்ணி ஆகணும். In-house na each thing you can decide. And of course, it's a huge big process. It is not as simple as it is told across. In-house in-house is very, very tough. But it can be done. It is being done in many places because dynamics of virus is very difficult. இப்போ நீங்க influenza virus எடுத்தீங்கன்னா இன்னைக்கு ஒரு influenza இருக்கு நாளைக்கு அதுவே வேற மாதிரி ஆகி it gets differentiated. So each time a manufacturer cannot bring out so many kits at a time. And we won't be using everything also. So adanale in-house is extremely important for the dynamics, but it is fairly a difficult process. Right? So these are the things where you guys can do a lot of research. Okay. Now, we go on to the types of ELISA. So again, this is what I said. This we go on. We have 4 types. Can we name the first one? Direct. So I think we go to the next slide. So we have the direct, the indirect, sandwich and the competitive. Excellent. So direct வந்து ரொம்ப cool. Antigen இருக்கு. You add the primary antibody அப்படினா antigen for the known disease ஏற்கனவே ELISA plateல இருக்கு. அந்த patient அந்த disease இருக்கா இல்லையா அப்படினு சீரம் ஆட் பண்ண போறோம். So அந்த antibody tag பண்ணிடுவோம். Tag பண்ணினோனே கீழ வந்து அது அட்டாச் ஆனோனே அந்த tag release ஆகி we see it as positive. It is as simple as that. So that's called the direct ELISA. Yes, ஒரே ஒரு step தான். But that is very, it's not very easy to do it. But specific அந்த antibody against அந்த antigen against மட்டும் தான் அதை use பண்ண முடியும். You can't use it, liberally on so many other ELISA plates. So it is not very famous, it is not usually taken as a common technique. Instead we do the indirect ELISA. Now this is direct ELISA. We go on to indirect ELISA. Now indirect ELISA is technique. Again you have the antigen, you have the antibody of the patient which has come and got bound. So that has become a complex. We are now going to add a tagged antibody. Eppadi antibody produce பண்ணுவோம்? I told you just now. Correct. So we induce it from an animal, or even now we have human antibodies. So that is tagged. So that is common because it's against all the human antibodies, right? So that common antibody can be used for many ELISAs. It need not be specific for one organism, because the organism is the antigen, the antibody for the organism has bound and it is already formed a complex. In the complex against our antibody podrom, tag the antibody. Is this clear? Yes or no? So we have a tagged antibody and that is called the indirect ELISA. Yeah? Because you are indirectly trying to guess what is the complex. Clear? Okay. Now we go on to, this is the diagram for the indirect ELISA. Then we go on to sandwich ELISA. It's just the English word sandwich, where we have the antibody on the ELISA plate. We have the antigen in the serum, over which we add the common antibody, which I have been talking about, tagged antibody. So this is a sandwich between the two. ரெண்டு ஆன்டிபாடிக்கு நடுவுல ஒரு ஆன்டிஜன் இருக்குது சாண்ட்விச். ஓகேவா? So அவ்வளவுதான். Nothing great about this thing, but this is a very usual technique that we use. This is called this is because it is finding out the antigen. What is P24? Is it an antigen or an antibody? Okay, we'll come to that a little later. Now we finished. Now this is the most important competitive ELISA. So we have found what is direct. We read what we tried deciphering what is indirect. Then we went ahead to see what is sandwich. Now we are going to see competitive. Competitive, there is a competition between the antibody which we have tagged and the antibody which is present in the serum. So we have antigen coated plates. We have the antibody of the patient. We are tagging the same kind of antibody. We are adding that also. So both the antibodies will try and come to fight for the antigen. But எப்பவுமே nature is great. So patientல இருக்கிற antibody எல்லாம் கடகடகடன்னு it will go and occupy many antigens inside the plates. So what we are adding, the tagged antibody, will be very less space will be available. So the lesser the value, the more the antigens. It is inversely proportional. Why? Because we have an antigen, we have an antibody from the patient which goes and covers almost all the antigens which are present in the ELISA plate. We have a tagged antibody which we are adding. They don't have space there. So they're all floating. அவங்க float பண்ணிட்டு இருந்தாங்கன்னா fluoresce ஆகாது. Once they go and settle on some place, they form a complex, அப்பதான் fluoresce வரும். So less the fluoresce, what happens here? More the antigens. So it is inversely proportional. So it is one of the best ELISA tests that we have. Is this clear? Right. ELISA க்கு நம்ம சொன்னது அந்த பேஸ்ல it is already there அப்படிலாம் சொல்றது வந்து entire protein can be coated. Then we went on to coat only the antigen. Then it was only the epitope of the antigen which was being coated. Now we have come to branch chain amino acids. Proteins are made up of? Okay. So branch chain amino acids. Now we have synthetic amino acids also. So the ELISA plate is becoming safer for us to handle. Right? ஏன்னா வாஷ் பண்ணும்போது நம்ம கவுத்தி இப்படி எல்லார் மேலேயும் தெரிச்சு எல்லாம் பண்ணுவோம். So all that can be done because it is synthetic now. That is why I spoke of P24, which is now a synthetic antigen also is available. Branch chain amino acid is also available. All that. Okay, so that is how the grades of ELISA have evolved. From proteins, we have now gone on to branch chain amino acids or even synthetic amino acids. Is this clear? Okay. Now, ELISA, it is quantitative or qualitative? What do we mean by that? What is quantitative? Amount. So, usually, ELISA is a qualitative test. Yes or no? It's only a qualitative test. Is it present or is it not present? Is what we are looking at. But there are now quantitative tests also available, which means it's actually a semi-quantitative test where we have standards given by the manufacturer. So in the standards, or optical density. So our end reading of ELISA is optical density. And how do we read that? We read it by an ELISA reader. Calorimeter is just a color that is extremely physical. Okay, so we don't use calorimeter. Of course, that is the base, that is the principle, but that is the color which can be read only by an ELISA reader, because we have filters specifically to tell us which range this color falls. Okay, so if I can go through quickly, do you know the steps of ELISA? Because I can go through it. I thought you all know about it. Okay, so first we try and add the patient's serum. We incubate for some time. Ella manufactured manual. You wash it 2 or 3 times. Then you go ahead and add the enzyme. Then you add the substrate and finally add the stopping solution and keep it inside a dark space. Okay, where there is no light. Then you take it to the ELISA reader and then you check the reading. That is how an ELISA is done. And in the manufacturer's manual, they would have given something called the cut-off value. Okay, so cut-off value ku mela irundha, it is positive. Cut-off value ku keela irundha, it is negative. Clear? Yes or no? Okay. Now, so it's just the cut-off value of the optical density. It is just the color, which is the predictive listing of ELISA. Okay. Now, if you have standards which are given, I told you about the quantitative ELISA, where you can, the manufacturer would have given you, first sample will have 1000 virus in this mule. If it comes, the OD value comes as this, it should come as 5. So this is just a temporary example. So if it comes as 5, you know the person has, that sample has 1000 virus, 1000 international units per mule. So number we can say, okay, it is near 1000. Right? So it becomes a semi-quantitative test. Yeah? But there are so many mistakes that can happen because it is the person who is handling the pipette. So the volume is extremely important. As I told you, it is just an optical density. It is just the color that is determined. If you leave it for a long time, you go for tea and come back and think, okay, stopping solution, stopping solution will make the entire ELISA plate colorless. Full color and you have to start all over again. So the timings which are given in the manual has to be adhered to. Right? Now, there is something called the washing buffer. Now, if they say 3 times you wash, you have to wash it for 3 times. You can't take a shortcut in those things because they have done so many tests before finalizing these steps. So you can't take a shortcut and say, I know, man, so many years I've been doing, let me do it 2 times. No, it won't work. Why won't it work? Again, because the concentration may vary. And if it varies, the color will become different. If the color becomes different, reader will show a higher value. Which means we are falsely calling a person positive. Okay, so this false positive can be avoided only if you stick on to the manufacturer's manual. என்ன எழுதிருக்கானோ verb pattern நீங்க போட்டுதான் ஆகணும். Positive control, negative control, blank. There is something called a blank which is used in ELISA. What is that blank? Whatever is the air which can cause a contamination, that will be blanked off. That will be minused from all the wells. Okay. Now next important thing is you have 96. So you keep counting 1, 1, 1, 2, 2, 2. That is how you add in the ELISA wells. Right? This is called a microtiter. Abdina evlo add pandrongradhu we will not even know. It's just in micromillimeter level. So when you lift it and see ayyo nama moonavathu add pannuma naalavathu add pannuma ayyo nadula yaro pesitangale indha mari pochuna gali நம்ம ரெண்டாவது add பண்ணோமா மூணாவது add பண்ணோமா add பண்ணலையா. So if this goes, entire color will go off. So the result will be redundant. அதனால தான் generally it is done with 2 people so that they keep account and we are also having account. We don't get distracted with cell phones, phone calls, tea, coffee, nothing of that kind when we're just adding this 96 wells. Adukapra you can enjoy. Then we have something called a mock sheet, which is very important, where you take a sheet with the numbers written on it. First well la enna add pannீங்க positive control, second well la enna add pannீங்க negative control. இத நீங்க போடணும். அப்பதான் ELISA ரீடர்ல போயிட்டு வெளியில வரும்போது you will know what each thing is meant for. So that's called a what sheet? Mock sheet. So that is very, very important. Right? So I think I will go on. I've skipped all this. Yeah. One more thing which I would love to add is something called the gray zone. I don't know if it is little too much for you, but anyway I will let you know. Gray zone na OD value ku kitta vara value. Okay va? Idu 0.5 OD value nu sonna 0.55 vanda adu positive or negative ah? Positive, yeah. According to my definition, anything above the cutoff value is positive. But 0.05 is the only amount which is higher than the OD value. So, this is called the gray zone. And this sample has to be repeated by a different principled ELISA. Abdina enna meaning? Where or principle? Yes, madam. So with direct ELISA, yes, madam. Not you, next one. Yeah, next to go. Yeah, tell me, what do you mean by 2 different principles of ELISA? Just now we spoke of it. You know? No. So it is, if we have detected an antibody with an indirect ELISA, we must do it with a sandwich ELISA. Just a different technique has to be used. Repeating the same sample with the same technique may not give you a big result. Adhirpi and the OD value adhe mari varum. Gray zone la varum. Gray zone is very important to deal with. You have to deal with it with a different technique ELISA. Is this clear? Okay. So now I finished with the possible errors. What is DNA? I thought somebody has already spoken to you last month about the DNA. What is DNA? Yeah. So there are purines, thymines. Idella paathutom illaya? Last month paathoma? Okay. So I won't go into all this. So why are we taking a part of this now? We have finished with ELISA. We finished agglutination, precipitation, flocculation. We've done ELISA now. Immunofluorescence, ELISA. We've come now to PCR. Right? PCR is full form is? I like it. Full form everyone knows. So it's a polymerase chain reaction. Polymerase chain reaction of the material. So that is the DNA which we are talking of. Right? So the DNA material la நம்ம எங்க இருந்து எடுக்க முடியும்? எந்த செல்ஸ்ல அந்த DNA இருக்கும்? Any guess? Can we take it from RBC? Why? So that's what I expected. So it's only the nucleated cell. Very good. So only the nucleated cells are the one which can give you the DNA. Okay. So we get the DNA out of it by either an ethanol separation or a heat separation and then go through a spectrophotometer. If ethanol separation, now we add the ethanol slowly and do a centrifuge and get the DNA material. The disease specific sequence. Sequence of nucleotides. Excellent. So purines, thymines, guanine and cytosine. Right. So in the naloda sequence, if you say HIV, you have a sequence which is very specific for the disease. And the primer number add panna porom. So that is the annealing when you are adding it. So the first part is denaturation, which takes place at 96 degrees, 97 degrees. Again, if it is in-house, you can adjust the temperature. If it is bought from the manufacturer, please stick on to the temperature. So once the denaturation, first the production of DNA, we saw it with spectrophotometer. We think that the DNA is great. So we are going to go ahead with the PCR. PCR la first step denaturation, second step primer add paniyachu. Ipo second step moda we are adding something called a master mix. And the master mix la we would have put the nucleotides, we would have added the primer, we would have added magnesium chloride. Everything will be there in the master mix. Because each time if we take it out and try and add, there will be a lot of confusion and there will be a lot of contamination. It is just the DNA that is getting expanded. Xerox copy of that DNA. Ninga tumminalum sari, hair lendu vandalum sari, kai lendu vandalum sari, anything will get expounded. Xeroxed. So, adanala it is very, it is mandate to do it in 3 different rooms. One is for extraction, one is for the whole process to take place, third is the documentation. Okay, so that will to an extent decrease the contamination. Anyway, after the primer is added, it will start annealing at a lower degree, 96 vanda, 53, 46, there are various temperatures allowed for annealing. Now once the primer has annealed, we go on to elongation. Elongation la nucleotides na potathanala from the primer end, 3', 5' end, it will slowly start expanding. 2 strands will become 4 strands, 4 strands will become 8 strands, 8 will become 16. Thank you. So that is what will happen. Now, but this is one of the, now we have some, once the PCR product is ready, 30 cycles mudinjiruchu, outflow, the DNA has undergone denaturation, annealing, elongation, ella Xerox copy form ayi or periya product vandiruchu. And the product, nammaloda product dana, nammaloda interest product dana kandupidikkiradhukku dhaan we do something called the gel documentation, where in the first line pathina it has got so many lines, which is a molecular weight marker. Every 100 base pairs there will be a mark. And the gel la நம்ம ethidium bromide போட்டிருப்போம், அதனால இது fluorescent ஆகும். So அந்த molecular marker first ladder போட்ட அப்புறம் நம்மளோட wells பக்கத்துல wells தெரியுதா, அந்த wellsல எல்லாம் we will add our PCR product. Okay, so once that is added and you leave it for electrophoresis, it will start traveling in the gel. Wherever it stops, that should correspond to the molecular weight. If the molecular weight of your virus and the molecular weight of the sample has matched, it means that that is present in the sample. Is this clear? No. Okay. So in the well, you are adding the PCR product. Right? First well molecular marker. So மேலேந்து பாத்தீங்கன்னா it will be 1000, 900, 800 அந்த மாதிரி 100 வந்து லாஸ்டா வரும். ஓகேவா? So now நம்மளோட சாம்பிள்ஸ்லாம் வெல்ல ஆட் பண்ணிருப்போம். அந்த வெல்ல இருந்து it will start migrating. Electrophoresis will happen. It will start becoming ஒரு இடத்துல வந்தோம் it will stop because அவ்வளவுதான் இருக்கு மெட்டீரியல். அந்த molecular weight ஓட மெட்டீரியல் வந்த உடனே where is it matching with the ladder னு பார்க்கணும். இப்போ HBV, say, and again an example, if it is 400 base pairs is for HBV, நம்ம PCR product 400ல வந்திருக்கா இல்லையா அப்படின்னு பார்க்க போறோம். வந்திருந்தது அப்படின்னா PCR positive. அவ்வளவுதான். Right? Yes? Okay. So that is about the molecular marker, the documentation. So now what are the types of PCR? Now I told you it is a DNA that we use. Can you name any RNA virus? HIV is an RNA. இப்போ அதில் PCR பண்ண முடியுமா பண்ண முடியாதா? முதல் ஸ்டெப்பே டீநேச்சுரேஷன். இதில் எங்கே டீநேச்சுரேஷன் வரும்? RNA இருந்தா வராது. So we do a reverse transcriptase, make it a double strand. So first step is not denaturation. There we make the RNA into a DNA and then go ahead with the same PCR. Okay. Then there is something called a huge primer proton vechukona. There are some diseases which may have big primers. Big more than 300 base pairs primer irundha. It may form a small loop. 80 irundhudu, CG irundhudu, thirukku or 80. So that will come and form a loop and come and get attached to the base DNA. If that happens, then we are not sure whether the entire primer has been attached or is it a loop. To prevent that, you have something called the nested PCR, which means the Suryan ke velichangara madhi primer ku innoru primer poduvom. That's called the nested. It's nested for the PCR, primer. So it's called the nested PCR. So on the mari there are plenty of them. Mutation, point mutation is there in thalassemia. Adula detect padradhukku you have point mutation PCR. There are plenty of them. So I don't think I need to go into that. One word on the real time PCR. Ipo na sonnadhu enadhu 30 cycles mudinjittu documentation pannina dhaan adu irukka illiyana theriyum. Idhan conventional PCR. Ipo ivlo nani pesinadhu conventional PCR. Okay. Whereas real time PCR la 10th cycle or 20th cycle you will know, 10th cycle you will know that the product irukka illiyana. You can stop the reaction. Because the moment the primer comes and settles over the strand, it releases a fluorochrome, quencher fluorochrome. There is something called two types. I think I have a diagram on that. Yeah. So you have a reporter gene and a quencher gene which comes up and gives a color. Again, fluorescent color. So the color and the dye நம்ம கண்டுபிடிச்சிட்டோம் 10th cycle அப்படினா great. இருக்குன்னு அர்த்தம். இல்ல வரவே இல்ல 10th cycle வரைக்கும் அப்படினா there is something wrong with our technique. Either you repeat the technique or it may be absent as well. So this is the real time PCR. So actually sir was talking about minutes la kudukanum result. Yes. So if that is the case, PCR is very very important. It gives a very fast result and very reliable result. More so, it is the gold standard for so many diseases. Okay. Now, but the primary problems, as I already told you about, is the primer problem, the hairpin problem, for which we use what PCR? Pardon? Nested PCR. Taq polymerase sometimes may be a problem. I think it is going a little above all your heads, so I will skip all these things. And we will go on to talk of. There are, as usual, limitations. The worst limitation being contamination in a PCR because it's just the DNA expression. DNA or a Xerox copy. And then the DNA push. So that is the problem. I will skip all this because of two things I think that you guys know. Now, microarray. There is something called multiplex PCR, which I forgot again, where we use as a screening test. 3 different viruses ஒரே PCR ல கண்டுபிடிச்சிடலாம். One PCR run ல you can check whether it is HPV, HCV, HIV positive by adding different primers, but we will know what is the infectivity. That's called multiplex PCR. Adoda expansion is the microarray. Okay, where you have a small sheet with a chip where you can lay all your valuable nucleotides of interest on each well. Okay, just see the plate there. It is so beautifully laid. Then you add the tagged serum with the antibody. It will go and get attached to whichever antigen is present. HBV na HBV oda poi sendukom, HIV na HIV oda sendukom. So அந்த மாதிரி சேர்ந்துறோம்னா it will give fluorescence. So on the DNA plate la, microarray plate la, you can recognize almost 100 and odd diseases at one time. But it has its own disadvantages. Again, I won't go into those details. But this is how it will look at the end of it. Fluorescent color will be different for each diseases. We will know how many serum samples are positive for that particular disease and how many of them are for some other disease as well. So that is about the microarray. Now we go on to these things. I will skip. Antibody production. Now I told you already, if we give our human antibody into any of the animals, it's going to produce antibodies. Yeah. So if it's going to produce antibodies against its own disease and against the human antigen, whichever we have given, it's going to be called the polyclonal. Abdina, polynale nariya. Right? But we want something very specific. If we blood group paakano, anti matuna veno. I don't want polyclonal and so many things coming in because it will be a mess there. So I want only anti-A. So I have to produce something called a monoclonal antibody. So that is what is the diagram here, where we introduce the antigen of interest into the mouse. It produces antibodies. That is laid on to the HAT medium, HAT medium, with the multiple myeloma cells. Here multiple myeloma? Cancer cells. Again I get from teachers. Myeloma is a cancerous thing. Ipo na sonnom Bence-Jones protein pati, myeloma pati, it's cancer. So it will start multiplying very fast. So that is going to be the monoclonal antibody. Now we use it is for the gene therapy. If a patient lung cancer, you can take out that gene, make antibodies against that gene as monoclonal antibody and give it back into the patient, which will act against that particular disease, that particular cancer. So that is the importance of monoclonal antibody. Is this interesting? I think given a lot in between, so now my talk sounds redundant, so I quickly go through it. The last word on HLA. HLA is something where the T cells presents to the B cell. Okay, now first so the T cell presenting to B cell is through the major histocompatibility complex, which is otherwise called the HLA. Now there are three types of HLAs: class 1, class 2 and class 3. I'll go to the next slide, where this is the one which I spoke, where the antigen is being presented. Most important part of the HLA. Now this is the methods which we find out. Adhe madridam HLA or antigen idukku plate Tarasaki plate, T-E-R-A-S-A-K-I, Tarasaki, which does not have 96, it has only 72 wells. In that you have again the antigen coated, you add the antibody. If the cells die, that means antigen is present. So that was the serology. Now we have the same principle used as PCR. Yes? At least listen to these things. PCR is next way of detecting the HLA. It has SSOP and SOP. I am not going into the details. So these are the ones. My hair is just like my dad. My eyes are just like my dad. All that is because of the HLA, which determines what do you bring from your forefathers or your genetic material. There are three, as I told you, three types, which is involved in tumors, in the genetic inheritance. Then you have the immune response, you have the immune memory, name it. Even the smell of the sweat is determined by the HLA, how important HLA is. Now I leave with the last question. Is fetus an external body or an internal body? Fetus is Papa, right? Yeah. Is it an external or an internal body? Internal? Fetus is somewhere in the uterus. Why should it be internal? Internally, you're doing a surgery and removing an internal organ from the mother. It is definitely an external. So let's get that clear first. Yeah? A baby is an external one inside the uterus. So why doesn't the mother say, I don't like it? Why is it a foreign body in my system? I'm going to throw it out. Don't tell me Thai person. That is totally different. But why is it happening? So I leave you with that question. This actually gives you the reply, but I would like you to search it and come back. Thank you so much.
Yeah, thank you for the class.
It's interesting session to come to a CME. Ask me also, because out of the class hours, they're sitting somewhere having a day and kind of enjoying. Doesn't matter. You can always come through sir. You can give him the questions and I can answer wherever possible, because sir himself knows a lot of stuff. And he himself will be able to answer most of the questions. Apriyum Tandi, you want to ask me only, Aprina, you can come through him and he can mail it to me and come back to you. Or as he said, you can all come in batches to our place and check what all facilities we have, so that definitely we can go a step forward, at least for your small projects and your big projects, whatever you feel like. Thank you.
Thank you, ma'am. We now move on to the next session. So please turn your attention to the screen for a short presentation on our Professor Dr. Saravanan sir. I now invite you to deliver the paper.
So good afternoon to one and all present here. At the outset, I would like to thank organizers inviting me to talk about one of the global crises and global top priority research on viral hepatitis, because the reason why the hepatitis B is incurable disease. As of now, we cannot cure hepatitis B like HIV, but we have a very strong medication and effective vaccines to manage and protect the entire population. So let us begin my talk to start with the burden of hepatitis B viral infection. Where are we stand as far as the country India is concerned? What's the global problem, global burden of infection, hepatitis B infection, and why we are still struggling to cure hepatitis B. So what's the reason? So what are all the barriers we face? Why we struggle a lot? Why we are not able to find a cure? So I think young brains, it's kind of the brainstorming sessions. You can come up with a novel, innovative idea how in future you can have a curative research on this particular global research priority area of hepatitis B cure. So I have nothing to disclose, no financial commitment. My talk. So for another 30 to 40 minutes, my talk is going to cover the brief introduction about the burden of hepatitis B and the standard treatment currently available globally as well as in India. And in 2024, the March, the World Health Organization expanded the treatment guideline, because they have fixed the target. We call them as a ambitious target. So World Health Organization, உலக சுகாதார மையம் வந்து 2030க்கான ஒரு டார்கெட்டை ஃபிக்ஸ் பண்ணியிருக்காங்க as far as the hepatitis B is concerned. அதுல மூணு important thing. The diagnose first பண்ணணும், treatment பண்ணணும், and then prevent the liver disease progression. So first 90% of them should be diagnosed. That's the first and foremost important target to achieve by the year 2030. And then of those 90% of the diagnosed individuals, 80% should be on treatment. So by putting them on treatment, you can drastically bring down the viral load because they are the only source of the infection. If you are able to contain the virus, there won't be any transmission at all. So that's the second ambitious target. Third one, we have to prevent the death due to liver cancer, at least 65%. So the diagnosis, treatment, prevention of the liver disease. Okay. And then why we need, it's very clear because we have to find the cure, otherwise what exactly is going to happen, what WHO want the entire global community. In fact, the government of India also taken a strong initiative to prevent, in fact, the elimination of this hepatitis by the year 2030. Just imagine we are in 2025, just 4 years left. So are we really going to achieve 2030 target is a big question. So where exactly we stand as far as the India is concerned, that is also I'm going to talk. And how exactly we can achieve the cure. So what are all the current curative strategies currently being explored in the world? உலகத்துல இந்த ஹெப்படைட்டிஸ் B-ஐ கியூர் பண்றதுக்கான என்னென்ன தீவிரமான ஆராய்ச்சியெல்லாம் பண்ணிட்டு இருக்காங்க. ஏன்னா டைம் ஃப்ரேம் ஆல்ரெடி ஃபிக்ஸ்ட் 2030 டார்கெட். நம்ம வந்து 4 வருஷம் தான் இருக்கு. ஸோ என்டையர் வேர்ல்டுமே பயங்கரமா அது முயற்சி பண்ணிட்டு இருக்காங்க. And then how do we achieve and clinical curative strategies? என்னென்ன இதெல்லாம் அடாப்ட் பண்ணலாம். So எப்படி பண்ணுனா how we can achieve the ambitious target by 2030. The whole talk of my presentation is going to be in and around this. So let's see the global burden. It's very simple. As you can see here, the major burden of chronic hepatitis B, as you can see here, the 254 million people currently infected with hepatitis B. They are all chronic HBV infected. That means they are infected having the virus beyond 6 months. So whoever is HBsAg positive beyond 6 months, we call them as chronic HBV individuals. So 254 million people currently they are carrying the virus. They are all infected. We cannot cure them, but we have the drug to manage. But if you look at this, this is really very pathetic situation. Of those 254, this is the recent data. Just 13% of them have diagnosed and know their status, of which 3% of them on treatment. So 254 million people, 13% தான் diagnose பண்ணிருக்காங்க. They know their status, but rest of them are not aware they are infected with hepatitis B. They are all knowingly or unknowingly transmitting the virus to others. If you look at the global epidemiological pattern, majority of hepatitis B infection is mother to child. So pregnant mother transmitting the virus to the baby. This is the most commonly observed transmission pattern as far as the hepatitis B is concerned. That's the reason why in all antenatal clinic, the screening for surface antigen has become mandatory, including India. If someone is pregnant in first trimester, there are certain set of parameters to be screened prior to the delivery. So first trimester 3 மாசத்திலே HIV, hepatitis B, C and syphilis things எல்லாமே நம்ம pregnant women க்கு பண்றோம். அது பண்றதுக்கான ரீசன் என்னன்னா hepatitis B மதர்ல இருந்து குழந்தைக்கு prevent பண்ணலாம். If you detect someone is positive in the first trimester. So we have a strong medications to prevent the transmission of virus to the baby and also to take care of their liver damage as well. So அம்மாவையும் protect பண்ண முடியும், transmissioனையும் protect பண்ண முடியும். Why we are very keen? As I mentioned the epidemiological pattern, more than 70% of the transmission of virus is mother to baby and early childhood. So that's the reason why we have strengthened antenatal clinic screening for hepatitis B. Okay. So this is another problem for hepatitis B. If you look at the global burden of HBV, 80% of chronic HBV infected individuals are low and middle income countries. So it would be very difficult to to implement any new novel innovative kind of curative strategy because the burden of the cost. So it's very difficult unless the big the sponsors like WHO and major funding agencies coming forward to sponsor would be very difficult. So what we can do? But vaccine is 100% protected, almost 100. There are some exceptional. In spite of having 3 doses, some of them are still susceptible. That's exceptional. I don't want to talk. So, despite having a 100% good, protective, effective vaccine, the coverage is 43%. So, not even 50%. So, remaining those who have not vaccinated are still susceptible for hepatitis B. This is what the current situation. And if you look at the burden, global burden, where are we exactly after China? India is in second position. So hepatitis B is endemic. We are falling in the intermediate zone. If you screen general population, you can detect a 3 to 4% HBsAg positivity. So this is what the current situation. But some research data from our own country projected that, say for example in Andaman Nicobar Islands, you can Google it, the 23% positivity. Just imagine if you screen the tribal population, out of 100, 23 of them are going to be positive. So this is the situation. So overall, I am telling the country per se, 11 million people. So 11% means nearly 40 million are chronic HBV infected individuals from India alone. Okay. So this is the biggest problem as of now. Why it is not moving? Oh. What is it? It's going, I think, I don't know. So thing is, so the next slide can, no. Okay. I can talk. So thing is, imagine every minute, just imagine, every minute 2 persons are losing their life. They are dying due to viral hepatitis. So WHO done a beautiful study. They have compared important infectious diseases, HIV, TB, malaria. They have calculated the mortality since the year 2000. So they could see some flat line of HIV, malaria and TB. So liver disease is the only disease, especially for hepatitis B related liver disease, is gradually increasing since the year 2000. So what they have said very clearly, unless we take action now, what will happen in 2040? 2040ல என்ன நடக்கும்னு WHO சொல்லிருக்காங்கன்னா, the entire world HIV mortality plus malaria mortality and tuberculosis mortality, all 3 mortality put together, you will get some number. That number is going to be, outcompete. This liver disease is going to outcompete the cumulative death of all 3 major infectious diseases. So how serious the problem of hepatitis B? So we have to take action now itself. Then only we can achieve 2030 target. So this is a beautiful picture. You can Google it. I just want to emphasize the key steps involved in hepatitis B. Hepatitis B is a DNA virus, but it's tricky to understand the core molecular content of the replication. Though it's a DNA virus, it replicates through RNA. Hepatitis B DNA virus இருந்தாலும் replication வந்து RNA க்கு போயிட்டு then DNA க்கு வருது. You know the problem whenever we get this kind of reverse transcription, there will be some miss that synthesis, because the reverse transcriptase enzyme is very poor in editing processes. It cannot make any correction. So if something goes wrong at the time of replication, reverse transcriptase cannot correct it. So that's the reason we see tremendous amounts of the quasi-species in RNA viruses. Reason why? Because they cannot make the correction. But DNA virus can able to edit. But this is falling in between. Though it's a DNA, it's going to replicate via RNA. So that's the reason we see a lot of mutants as well. As you can see here, entry, can I have a pointer, Mardana? No? Okay, no problem. Protein is the receptor in our liver cell where the virus is going to sit. That's the initial attachment in our liver. And then the core content of the virus delivered in the cytoplasm. And then the unique characteristic feature of hepatitis B is the partially double-strand DNA. Just imagine, one DNA is complete, another DNA is incomplete. So two double-strand DNA, circular, but it's not complete. So we call them as a RcDNA, relaxed circular DNA. So that relaxed circular DNA exported into the nucleus of hepatocytes, where with the help of our host enzyme, it will become a complete double-strand DNA. So it obviously requires the host enzyme in order to make its own DNA into complete double-strand form. So the problem here in hepatitis B, once this stage is attained, the persistence of hepatitis B in our body in two forms. So virus can persist in CCC DNA, covalently closed circular DNA, and some part of the genome can also be integrated in our host chromosome like HIV. So there are two forms of viral persistence in hepatitis. It's a really holy grail for the cure for hepatitis B. As of now, till now, we don't have any medication to touch, not even to touch the DNA of the infected individuals. That's the reason. So once someone is infected, persistence is established in an infected hepatocyte nucleus, it's forever till death. Okay, so that's the main culprit for the hepatocellular carcinoma. Though it will take time for few decades, but it can also induce a lot of oncogenes to progress the disease. Okay, so that's the reason. But one beauty naturally God has given. So morning, madam on the immunology class nalla eduthaanga. Hepatitis B la, you remember, this virus is a non-cytolytic virus. You have a cell. Virus is going to infect. That virus is not going to lyse the cell. But we see infected individuals, the transaminase, ALT, AGPT, AGOT elevation. So that indicates the inflammation of the liver. So inflammation of the liver is due to immune response to virally infected hepatocytes. So virus ஒன்றும் பண்றதில்ல, நம்மளுடைய body immune response தான் அந்த elevation of ALT க்கு reason. Okay, so whenever we see the transaminase elevation, liver inflammation is due to our immune response. As you can see the bottom left side acute, adults more than 18 years, 100 people infected with hepatitis B. 95% of them can resolve the infection. Just imagine, 95% of the adults can resolve the infection even without taking a single antiviral drugs. We have got such a strong CTL immune response, innate immune response. Adaptive, you have two different T cell and B cell immunity. So naturally we have got a strong immune response to tackle the early acute stage of viral infection. But tiny proportion. So that's the tiny proportion of the individual, the 5%. Out of 100, 5% of the adults not able to clear the virus within the 6 months. They are going to become a chronic hepatitis B. Those 5% only, we have a 254 million in the world now. So just imagine, actual infected is 2 billion. 2 billion people infected, resolved majority of them, 5% only can carry forward, can carry the virus throughout the entire life. This is applicable only for the adults. Classify them in different phases. It's a bit of complicated. Gastroenterology is, struggling like anything to identify what stage they are. If someone is positive beyond 6 months of surface antigen, the immediate next step is we have to classify them into what stage they are. Then only they can take the clinical decision. So how? Broadly we can classify based on E antigen positive and E antigen negative. The virus is there in the liver. So we cannot take the biopsy to understand the virological characteristics feature. So we have a surrogate marker in the blood. So simple ELISA, morning madam explained very beautifully but nicely about different types of the ELISA. So by adapting the ELISA methodology, you can do the HBeAg. So envelope antigen is a surrogate marker to understand the active viral replication. If someone is positive for E antigen, they are having hepatitis B, the virus is actively multiplying. So that's the danger. So we don't treat all hepatitis B infected individuals. We don't have the treatment guideline says very clearly, not all of them are eligible to go for treatment. So there should be some eligibility criteria. That's the reason why if someone is positive for surface antigen, we have to assess their phase, the chronic phase of the infection. As you can see, those two stages highlighted, they are infected and also they are presenting the elevation of ALT. That indicates their immune system fighting against the virus. So that's the stage where we have to start the antiviral drugs. That's what the guideline says as of now. This is what currently being used, even our Indian Gastroenterology Society as well. So this is how we treat the hepatitis B infected individual. This is what the 2024 guidelines. So 2024 guideline says very clearly is that, so in order to achieve 2030 ambitious target, so all the infected individuals, 80% of them are eligible to go for treatment. So such a way that they have, developed with the guideline, the 2024 expanded guideline. So we are very strong in that. And these are all the different medications, very old medications, interferon and nukes. That is what currently being used. But unfortunately, the currently prescribed drugs to treat hepatitis B is not able to do anything with that DNA. You got it? So you can suppress the virus, but you cannot remove the virus from your body. That's what I say. Okay? So, I'm not able to, if anyone can, yeah. So why we need cure? But that lifelong, treatment is lifelong. So it's very difficult. Any treatment with the lifelong is always the adherence associated with that. So we have to have the curative strategy. So we should have some stop point for the treatment. So then only we can, programmatic approach I'm telling. So lifelong treatment is always associated with so many complications. That's applicable even for hepatitis B as well. If they are not, using the drugs as prescribed by physician, obviously the virus can develop the resistance. So then it's a completely different epidemiological pattern. So just imagine, the transfer of resistant virus to the community, because it's going to be another problem. So it's always better to have the cure. That's what my talk. So this is the fantastic meta-analysis done. So as I mentioned, majority of the infection, the huge burden of hepatitis B in, unfortunately, low in middle income countries. So whenever we see this kind of problem, the immediate next question is the cost. So without cost, nothing will move. So they have done a beautiful meta-analysis that cost is very effective. I mean, cost effectively we can approach the cure. So we can implement the mass screening, vaccination, treatment, and also the perinatal treatment as well. So pregnant mother to child transmission as well. So they have done a wonderful analysis. So it says very clearly that cost is not the issue. We can implement the cure strategy across the world, including India, but we are also in the developing country. So these are all the different drugs. So what I have shown that previous slide is a standard drug that has already shown that, demonstrated that we cannot touch the DNA. But there are plenty of the drugs, more than 40 different novel drugs which can able to interfere several important life cycle steps. So the drugs what currently we give is only to target reverse transcriptase enzyme. But all those drugs highlighted in this schematic diagram can tell you there are some other vulnerable steps of hepatitis B life cycle which could potentially be used as a target for developing curative drugs. That's what it have. You have this slide. I think some problem in this animation, I don't know. Okay. So the barriers is why the problem? We have two levels of the problem. So persistent CCC DNA, as I mentioned, and defective immune response. So whenever we approach the cure, we have to correct both immune modulatory approach as well as the virological suppression. So simultaneously you have to do both in order to achieve the cure. So this is again the problem. Another important thing is the whole scenario is in the liver. Liver is the very poor organelle to initiate T cell, B cell activation. Just imagine. So how clever the virus. So the virus can exploit that condition. So it is the poor site for the immune response. Why? Too much of IL-10, too much of TGF-beta, and too much of Treg. All those things not allowing T cell, B cell interaction. So immunosuppressive agent. Lot of immunosuppressive agent in the liver. In generally we call liver as immune tolerogenic organ. That's not the good organ for immune response. That is where all the problems we have in hepatitis B infected individual. So virological problem, immunological problem, the place where exactly virus is going to give us trouble. That liver organelles also, it's another problem. So all those things you have to tackle. So as far as the cure, we should concentrate on both, all three. Persistent DNA, both forms of DNA to be removed, and also subviral particle. So surface antigen, if you measure one virus plus 999 or empty capsid. Just imagine. So 1:999. So we measure in ELISA surface antigen, that's all subviral particle. They are not infectious. How clever the virus? They are unnecessarily producing lot of antigen which are going to be neutralized by your T cell, B cell. There is no use. The virus is only one. So virus can easily escape. That's the reason why we call subviral particle as an immune decoy. It's a stealthy virus. Thirtuthanamana virus. How can easily avoid this? Because producing unnecessarily antigens. That antigen, தேவையில்லாத antigen போயிட்டு T cell, B cell fight பண்ணுது. But ஒரு virus தான் அதுல problem. That virus can easily escape. So this is how virus can adapt immunotolerogenic organ. Organல problem இருக்கு. T cell, B cell already reaction perform பண்ண முடியல because immune suppressive environment, virus permanent reside ஆயிடுச்சு DNA. As of now we don't have any drugs to target the DNA. Two forms of DNA. ஒரு DNA ல பிரச்சனை இல்லை. CCC DNA host virus ல integrate ஆகிற DNA. So several problems associated with that. So this is what? How exactly the virus can escape? This is how. So by producing enormous amounts of unwanted antigen can easily escape the virus. So the scientists, the research data have shown very clearly is that of those different proteins, this is the only protein. Surface antigen is the main culprit. There's immune decoy. Okay, it's helping the virus. So whatever the parameters we do for the assessment, the E antigen positive, E antigen negative, completely different virological characteristic feature. It's very difficult to understand. If you Google it, you can get the big, serological pattern. You just go through the serological pattern. It's very difficult to interpret because we have a different types of antigen, antibodies to make the final clinical decision. So many parameters are not favorable for the currently approved drugs. These are all the problems. So different types of cure. Whenever we do, we don't have the cure as of now, but we can try. So scientists have demonstrated some level of the cure. That's called partial cure and functional cure. Just imagine, functional cure, HbA1c undetectable, DNA undetectable in your blood. If someone is positive with the functional cure, if they give the blood, your ELISA can give you the negative. So that means the protein expression is silenced, but your DNA still persists. That's called functional cure. But it's the terminology is ongoing debate among scientists. Some people claim that not to use functional cure, it has to be called as the remission, virological remission. But let it be. That last one is the complete cure. That's what we are trying to achieve. Complete cure means you have to get rid of both forms of the DNA, integrated as well as CCC DNA. It's a big question. It's ongoing research. As of now, we don't have any remedy for this. So we have to approach both. So by reducing virological, are we able to achieve immunological? Not really. So we can somehow able to manage with the novel drugs. These are all the different novel drugs, capsid assembly modulators, nucleic acid polymers, all those things have shown very promising results as an individual component. But that is alone not sufficient enough to achieve cure. So there are plenty of the drugs in different stages of the clinical trial across the world. But it's not sufficient enough to achieve the functional cure of hepatitis. There are plenty. You can Google it. But what my statement is here is, and you have to do both simultaneously. Try to suppress the virus as low as possible. At the same time, you have to activate immune response as well. So immune response, by reducing this, we are not able to achieve adequate amount of immune reconstitution. So we should have some kind of strategical, algorithm in order to efficiently, induce immune response. That can be achieved. This is the same diagram. This is a gold standard. Just imagine 100 individuals, adults infected with hepatitis B, 95% of them naturally resolve. That is the blueprint we have. So that means our immune system can able to clearly, remove out the virus. So something we have naturally. So we have to tap that particular point. So that's the blueprint we have as of now. That's what currently is being explored. How come 95% of adults can able to clear the virus? Only 5%. So this is the blueprint for the immune-mediated approach of the cure. Okay, so that host immune response can be tackled by 3 different strategical approach. We can stimulate antiviral drugs by adapting TLR, it's an innate immune response, and also the new cells, and rescue ectototal. Some cells can unnecessarily express PD-1. So PD-1 can suppress the immune response. So by adding the antibody to neutralize the PD-1, we can automatically enhance the immune response of the T cell. That's the rescue of exhausted cell. So several, PD-1 is one of the best immune exhaustion marker, and other markers as well, the TAM you have, SIM you have, there are several markers to assess the exhaustion that can also be targeted. So these are all the different types of things. Vaccines is going. So currently people are exploring multiple combinations. So now we have more than 40 different types of novel. We don't want to do individually. Now they do mix and match. So they mix everything. This is what the currently recommended strategical approach of cure. We have a very good drugs to interfere with the life cycle, and we have a good drug to reduce the antigen load and immune modulation. So you have to find the best combination of all 3 mixed cocktail to achieve this. This is what currently being explored in the world as of now. So coming to my concluding statement, the countdown to the 2030 elimination is very clear, but the speed, nama pannakudiya research speed is not sufficient enough. So we have to, move our research in a fast track manner. So the main problem of hepatitis B is the funding. So we need funding to explore a lot of the basic science and then translational research. And the problem, the main culprit is CCC DNA and also the integrated DNA. As I mentioned, the different stages of the viral chronic hepatitis B, each and every phase has got unique characteristic feature. So now this is another problem, whether we can have a gold standard set of panel to assess the chronic infection? No. All the phases are not equal. So if you identify, if you come up with one particular marker, may not be reliable for other chronic phase of the infection. So whatever the drugs you have, that has to be validated for every stage of the chronic hepatitis B viral infection. Okay? So this is the take-home message. So it's time for action because almost nearing 2030. So know your status. So this is the question. How many of you vaccinated for hepatitis B, completed all 3 doses? Can you raise your hand? How many of you? 3 doses of hepatitis B vaccine. Come on, only few. Why not others? Okay, so those who have raised, the next question is, have you assessed your titer, antibody titer? When did you measure? That's the important. Because just mere completion of 3 doses is not sufficient enough to claim that you are protected against hepatitis B unless you assess the antibody titer. That should be the minimum of 10 milli international unit. If you don't have, you should go for the boosting. So my concluding statement is that just mere completion of vaccination is not equal to immunization. Just 3 doses of the completion of vaccine is not equal that you are immunized against hepatitis B. 3 dose complete பண்றது மட்டும் பத்தாது. அந்த 3 dose மூலம் நம்ம பாடியில sufficient amount of antibody produce ஆயிருக்கான்னு பார்க்கணும். Otherwise, it will be very difficult to say that you are protected against hepatitis B. In spite of having 3 doses, if you are not able to produce sufficient amount of titer, still you are susceptible. Still you can acquire the infection of hepatitis B. So, protect you first. Test, treat, vaccinate. Protect you, your family, your nation, and the world. If we take this kind of oath now, then obviously it will be possible to achieve 2030 target of 90% of the diagnosis, 80% of the treatment, 65% of the prevention of the death due to liver disease. This is what the ambitious target set by the global health community. This is applicable for even India as well. Okay? So I would like to acknowledge some of my mentors where I have undergone intensive training on the various hepatitis B, C, HIV sequencing, bioinformatics, and core molecular techniques. So this is my mentor, Professor David Smith, UCSD. Still I am in touch with him. Soon we are going to have a hepatitis B curative research. It's in UCSD. And she is the one proposed the first HIV cure in baby. She is Deborah Prasad in Johns Hopkins, where I have undergone all hepatitis B and C related sequencing of NIH funded clinical trial. So, and then Chloe Theo, she is the fantastic researcher in Hopkins University. She is the head of ACTG, Adults Clinical Trial Group, core virology head. So, she is also my collaborator. So, I'm going to show some of my publication. All those publications you could find my mentors. So it's all freely available in the PubMed. Just go through the methodology component. If you are really interested, as our directors are mentioned in the morning, so we are interested to have a collaborative research with this institute. And also, he is the fantastic scientist, Stuart Ray. He himself developed some software to identify the recombination. So I've been trained on this particular software for identifying recombination breakpoint. And the twin lady from Johns Hopkins again, Professor Alex and Susan. And as you can see here, Gene Moore is just behind that. You can see the Niagara Falls, the border between United States and Canada. So he is the head of the Global Viral Research Network. So where I have undergone the bioinformatics training as part of NIH clinical trial. And the Cure Academy, 2 consecutive years, I have been elected as an international research fellow for HIV and HPV cure. The first one is 2018 and 2019 and Mexico conference. So these are all the stalwarts of curative research of HIV, hepatitis B in the United States. They're all the big shots in NIH clinical trial. I'm in touch with them for developing cure protocol for our country perspective. not the international. We have to think about the Indian context of hepatitis B. It's completely different natural history as far as the virus is concerned. And then this is the new society we have formed in Chennai, Infectious Disease Society. We had successfully conducted February 2025 conference, initial conference here in Chennai. These are all the key pillars of this society, and I am one of the advisory committee member of this. And these are all some of my hepatitis related publications freely available in PubMed. You can. And now very fortunate to have Professor A.R. Venkateshwaran, former MMC director. He is the head of the Department of Medical Gastroenterology in Savitha Medical College Hospital. Now I am closely associated with him. I think he is also interested to develop a lot of the clinical oriented curative strategy for hepatitis B. So we are working on a new protocol for this. Last but not least, the left side, Professor S.P. Tyagarajan, my mentor, PhD guy, and the Professor Suniti Solomon, the late Professor Suniti Solomon, she's no more. She's the one documented the first HIV in India. You know Professor Suniti Solomon? First HIV India la irukku அப்படினு report பண்ணது 1986. When she was the professor and head of microbiology in MMC, 1986ல HIV இந்தியால இருக்குன்னு முத ரிப்போர்ட் பண்ணது எங்கன்னா சென்னையில தான். That is by Professor Suniti Solomon. So where been associated with her almost 2 decades. With her support, I have undergone all the international trainings. Of course, the cost is the key component of international training. These are all some of the funding agency helped me to undergo intensive training through NIH. These are all the different funding agencies helped my international training. So with this, I thank this opportunity. Special thanks goes to my Anna Marudhu Ramachandran, Anna, my senior. When I was doing my post-graduation, I learned a lot from Marudhu Anna. So you guys are really fortunate to have him. He is fantastic researcher. I know since beginning of his post-graduation itself. So he was very active when we were doing the post-graduation at Virudhunagar Microbiology College. Thank you, Anna, for inviting me to share some of my experience with the budding brains. It's kind of brainstorming session. So you guys can come up with the new novel innovative idea for the future. This can go for another few decades, the research for finding the cure for hepatitis B. So with this brief, I think I thank everyone for your patience. I would be happy to take some questions if you have. Really. Thank you.
Come to the end. So I now request Dr. Meera Devi Sri ma'am to give the vote of thanks.
Sorry for that. I forgot to mention Meera madam. I was her doctoral committee member when I was in Vayachika. I was the committee member, right? Yeah. So another, active researcher. So you guys are really, fortunate to have this kind of team. So you can explore anything. You can compete the global standard of research, actually. So I will be happy to have a collaborative research. We have got a lot of cohorts. I think I've briefed about everything to Marudhu Anna and also the director, sir. Okay? So,
Good afternoon to one and all present here. I extend my heartfelt thanks to Almighty for conducting the one-day seminar in a successful manner. It is indeed a great honor and privilege for me to propose the vote of thanks on this wonderful occasion. On behalf of the organizing team and the National Institute of Siddha, I wish to thank each one of you individually who made today's event a grand success. First and foremost, I would like to thank our Honorable Director in charge, Professor Dr. G. Sendanen sir, Dean in charge, Professor Dr. M. Meenakshi Sundaram sir, Dean Research in charge Professor Dr. G.J. Christian sir, and our HS in charge Dr. P. Manjari ma'am, as well as our BRS sir, Sandidel sir, for gracing this inauguration ceremony and for their constant encouragement and support. A special thanks to Dr. M. Madhuramachandran sir, Metabolism Division, for his warm and insightful welcome address, and to Tamil Hobia Biochemistry Department for anchoring the event with such enthusiasm and grace. We are deeply grateful to our distinguished speakers of the day. Dr. Pushkala ma'am, Professor, Department of Immunology, the Tamil Nadu Dr. M.G.R. Medical University, for her enlightening session on a sneak peek on immunological techniques. Her insights gave us a broader understanding of complex techniques in the diagnosis, I guess. And Dr. S. Saravanan sir, Professor, Department of Virology, Sabita Institute of Medical and Technical Sciences, for engaging the session on HPV cure paradox. Sir, your presentation was thoughtful, thought-provoking, and highly informative. We are very smart to okay. We are sincerely appreciate the time and effort you took to join us today. Your involvement reflects a strong spirit of collaboration and learning that we all strive for in academics. I also thank UG first-timers profession students, especially Ashika, Ashmita, Danya, Kavya, Renisha, and Siti, for creating such a wonderful AV about the speakers today in a short span of time. Last but not the least, I thank all the participants, faculty members, technical staffs, photographers, and each and every members of the institute who worked tirelessly behind the scenes. Thank you all once again for your presence, participation, and support. Let us carry forward the knowledge and insight we have gained today and apply them meaningfully in our respective fields. Thank you. Let's stand for let us rise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
எனர்ஜி அந்த சைக்கிளில் நம்ம எந்த ஒரு இன்டர்ஃபரன்ஸும் கொடுக்கக்கூடாது. ஏன்னா பூமிக்கு இந்த ஆகர்ஷண சக்திங்கிறது பேசிக்காகவே இருக்கும். அப்போ என்னன்னா எந்த வித ஒரு டிரான்ஸ்ஃபர்மேஷனும் நமக்கும் பேஷண்ட்டுக்கும் நடக்கக்கூடாது. ஃபார் எக்ஸாம்பிள் இப்போ பேஷண்ட்டை தொடுறோம், நீங்கள் என்ன நினைப்பீங்கன்னா நல்லா சாப்பிட்ருக்கோம், எனர்ஜெட்டிக்காக தான் இருக்கணும். பேஷண்ட்டு நான் சொல்கிறது இதில் தான் இருக்கு. நீங்கள் பார்க்க போகிறது எல்லாமே பேஷண்ட்டை தான், நார்மல் பர்சன்ஸ் கிடையாது. பேஷண்ட்டுன்றப்போ அந்த நெகட்டிவ் எனர்ஜி ஆகிறாங்கிறது வந்து அவங்களுக்கு டிஃபால்ட்டாக கொஞ்சம் டாமினண்ட்டாக இருக்கத்தான் செய்யும். நீங்கள் சும்மா உட்காந்து நீங்கள் நாடி தான் பார்க்க போறீங்கனாலும் 4 பேஷண்ட்டுக்கு மேலே 5-6 பேஷண்ட்டு கை வைக்கும்போது உங்களுக்கு பசிக்கும், எனர்ஜி ட்ரெயின் அவுட் ஆகும். புரிஞ்சதா? நம்ம ஒன்றுமே பள்ளியை உட்காந்து தான் நாடி பார்க்குறோம், ஏன் இவ்வளோ எனர்ஜி ட்ரெயின் அவுட் ஆகுதுங்கிறத நீங்கள் பார்க்க ரியலைஸ் பண்ணுவீங்க. ஸோ அப்போ அதனால தான் செருப்பு போடலைனாலே ஆட்டோமேட்டிக்காக நம்மளை தெரியாமல் நடக்கக்கூடிய அந்த எனர்ஜி டிரான்ஸ்மிஷன்ஸ் எல்லாமே பூமி வாங்கி. ஓகே, இதுவும் ஒரு ரீசன். தெரிஞ்சு பின்னக்கூடாது. 4வது விஷயம், நம்ம நெட்டை வாங்குதல்ன்ற விஷயம் நம்ம பண்ணணும். பேஷண்ட்டுக்கு நாடி பார்க்கும்போது சொடக்கு போடுறோம் இல்லையா, 5 விரல்களையும் 5 வாயுக்கள் இருக்கு இல்லையா, பிரைமரி வாயுக்கள், அதாவது முதன்மையான வாயுக்கள் 5, அதுக்கான உப வாயுக்கள் 5. ஸோ தச வாயுக்களையும் நம்மளால அந்த 5 விரல்களை நெட்டை வாங்குதல் மூலமா ஸ்டிமுலேட் பண்ண முடியும். ஸோ இழுக்கலாம், இழுக்கிறீங்கன்னா ஸ்டிமுலேஷன்ஸ், மடக்குறீங்க, இப்படி சொடக்கு போடுவீங்கன்னா கட 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 கடன்னு சொடக்கு போடுறீங்கன்னா அந்த வாயுக்களோட இயக்கத்தை தடை பண்றீங்கன்னு. அதனால தான் சொடக்கு போடக்கூடாது. நரம்புகள் பலவீனமாகும் சொல்லுவோம் இல்லையா, எப்படின்னா அந்த வாய்க்கள் தானே இயக்கம் எல்லாமே, வாய்க்கள் வழியாக தானே எல்லா ஆற்றலும் போய் சேரும். அது தடைப்படுத்துறப்ப நரம்புகள் ரொம்ப சீக்கிரமாக பலவீனமாகுங்கிறதுனால செய்யக்கூடாது. அடுத்து criteria patient உடைய heart லெவலுக்கு கீழே கை இருக்கிற மாதிரி பார்த்துக்கணும். இது பேசிக்காக எல்லாருக்குமே தெரியும். தூக்குனீங்கன்னா பல்சேஷன் குறையாது. ஸோ பெட்டர் இது எல்லா கிரைட்டீரியாலையும் மைண்டில் வச்சுட்டு நம்ம நாடி பார்க்க போகிறோம். ஸோ இது மேல் ஃபீமேல்ன்றதெல்லாம் தாண்டி நாடி பார்க்கறதுக்கான ப்ரொசீஜர் என்னன்றதை நான் செஞ்சு காட்டுறேன். நீங்களோட லெஃப்ட் ஹேண்டில் செய்யட்டுமா இல்லை ரைட் ஹேண்டில் செய்யட்டுமா, எது உங்களுக்கு வசதி? இதே தான் அப்படியே ஆப்போசிட்
சார். இது
இங்கே நிற்கணும். ஓகே, இப்போ ரெஸ்ட் ஜாயிண்ட்டுக்கு பின்னாடி கையை பிடிக்கணும் கரெக்டாக. சரியா? இந்த 2 விரல்கள் இருக்கு இல்லையா? இப்போ நான் ரைட் ஹேண்ட்லேயே கூட செஞ்சு காட்டுறேன். ஏன்னா லெஃப்ட் ஹேண்டில் செய்யறது சரியாக வர மாட்டேங்குதுன்னா இங்கே நமக்கு தேவை ப்ரொசீஜர் தான். இப்படி நான் பிடிச்சிக்கிறேன், தெரியுதான்னு மட்டும் பார்த்துக்கோங்க. இந்த 2 விரல்களை யூஸ் பண்ணி ஒவ்வொரு விரலையும் ஹோல்ட் பண்ணி சின்ன ஒரு சர்க்கம்ஃப்ளெக்ஷன் கொடுத்து லைட்டாக ஜெர்க் கொடுக்கணும். ஓகேவா? பிடிச்சி இழுத்து சொடக்கு வர்ற வரைக்கும் இழுத்துக்கிட்டே இருக்கக்கூடாது. இதனுடைய கான்செப்ட் சொடக்கு சத்தை கேட்குற வரைக்கும் இருக்கணுங்கிறது கிடையாது. தசை வாய்க்களை ஸ்டிமுலேட் பண்ணி தூண்டுறோம், அவ்வளவுதானே. சரியா? நான் ஒரு முறை செய்கிறேன், 2வது முறை நீங்கள் தான் செய்ய போறீங்க. ஓகேவா? ஹோல்ட் பண்ணிக்கிறீங்க. ஒரு விரலா 2 விரலுடைய சப்போர்ட்டை பிடிச்சி ஒரு சின்ன சர்க்கிள் 1. ஓகேவா? 2 3 4 5. உனக்கு வரல, நமக்கு கான்செப்ட் அது இல்லை, பரவாயில்ல. இது ஃபர்ஸ்ட் டெக். முதல்ல நெட்டை வாங்குதல் இருக்கு. அதுக்கு அடுத்தபடியா 5 விரல்களையும் மொத்தமா மடக்கி திரும்ப மொத்தமா நீட்டி உள்ளங்கைகளை ஹார்ட்டை நோக்கி சும்மா ஒரு 3 to 5 டைம்ஸ் சூடு பறக்க தேய்க்கணும். சும்மா லைட்டாக தேய்க்கக்கூடாது, வாமா சூடு பறக்க தேய்க்கணும். ஏன்னா வேகமாக தேய்க்கும் போது தான் அந்த வாயுக்கள்ன்றது இயங்காது. எல்லாமே அந்த கான்செப்டுக்காக தான். சரியா? தேய்ச்சி முடிச்சதுக்கு அப்புறமா ஷேக் ஹேண்ட் பண்ற மாதிரி கையை வைக்க போறீங்க. அந்த கை வைக்கக்கூடிய இடம் என்னன்றதுக்கான கரெக்ஷன்ஸ் காலையிலேயே நம்ம பார்த்துட்டோம். கட்டை விரல் வைக்கக்கூடியது பூது நாடி, ஆட்காட்டி விரல் வைக்கக்கூடியது சொர்க்கவாசல் நாடி. இதுல இருந்து கரெக்டா ரிஸ்ட் ஜாயிண்ட்ல இருந்து ஒரு விரல் தள்ளி வாதம், பித்தம், அடுத்தது கபம். ஸோ ஆட்காட்டி விரல், நடுவிரல், மோதிர விரல் 2யும் சரி 3 விரலையும் சரிசமமா வைங்கன்றது தத்துவம் காலையிலே மேடம் சொன்னது ஞாபகம் இருக்குன்னு நினைக்கிறேன். ஒன்னோட ஒன்று முரண்பட்டு வைக்காம சரிசமமா வைங்க. ரொம்ப கன்ஜஸ்டடாகவும் வைக்க வேண்டாம், ரொம்ப தளர்த்தி தள்ளியும் வைக்க வேண்டாம். டிஃபால்ட்டா 3 விரலையும் மடக்கி அடுத்தடுத்து வச்சிங்கன்னா எந்த லெவலில் இருக்குமோ அப்படி வச்சிங்கன்னா போதும். சரியா? ஸோ அப்படி பார்க்கும்போது இது வாதம், பித்தம், கபம். கடைசியாக இருக்கக்கூடிய சுண்டு விரல்ன்றது ஆத்ம நாடி சொன்னாங்க இல்லையா? இப்போ ஆத்ம நாடிங்கிறது நம்ம எப்போ பார்ப்போம்னா லைஃப் எந்த லெவலில் இருக்கு, அதாவது என்னன்னா அவருடைய ஆயுள் நிர்ணயம் அப்படிங்கிற ஒரு விஷயத்துல வரும்போது தான் இந்த ஆத்ம நாடிங்கிறது தேவைப்படும், மற்ற சமயங்களில் தேவைப்படாது. ஓகேவா? இப்படிதான் நாடி பார்க்கணும். சரியா? பார்க்கலாமா? ஓகேவா? இப்போ கீழே போய் நீங்களுமே ப்ராக்டிக்கலாக பார்க்க ஆரம்பிங்க. நான் கமெண்ட் பண்ணுறேன், பண்ணுற மாதிரி கரெக்டாக. ஃபர்ஸ்ட் என்ன விஷயம்னா 1 to 1 நீங்கள் நிற்க போறீங்க. பேஷண்ட்டாக இருக்கக்கூடிய நபர்கள் கீழே உட்கார போறீங்க. பார்க்கக்கூடிய நபர்கள் ஒன்று பண்ணுங்க, ஒன்று நிற்க முடியும்னா பாருங்க. ஏன்னா நாடி பார்க்குறப்ப நிறைய தப்பு வரும். ஸோ நின்று பார்க்க முடியும்னா பார்த்துருங்க, அப்படி இல்லைன்னா உட்காந்துருங்க. ஆப்போசிட்ல சேர போடுங்க. உங்களுக்கு எது கன்வீனியன்ட்டோ சரி. ஈஸியாக
ஓகேவா? ஸோ ஃபஸ்ட்டு பல்சேஷனில் நம்ம யூஸ்வலாக பல்சேஷனில் ஒரே நேரத்தில் மூணுமே துடிக்கும் இல்லையா? வாதகத்தகம் மூணுமே துடிக்கும். ஆனால் இங்கே அப்படி இல்லை. ஃபஸ்ட்டு பல்சேஷனில் பித்தம் துடிக்குதுன்னா அது அவருடைய முதல் துடிக்கும். ரெண்டாவது பல்சேஷன் வரும்போது பித்தத்துலையும் வாதத்துலையும் சேர்ந்து துடிக்கும். பித்தவாத தேகின்னு அர்த்தம். ஓகேவா? மூணாவது பல்சேஷன் தான் வாதகத்தகம் மூணும் சேர்ந்து துடிக்கும். தேர்டு பீட்ல தான் ரொம்ப கஷ்டம். சொல்லிக்கிறது ஈஸியாக சொல்லிட்டேன், இதை கண்டுபிடிக்கிறது ரொம்ப கஷ்டம். திணறி. ஸோ இன்னொரு விஷயம் நான் சொல்ல மறந்துட்டேன். நாடி பார்க்கும்போது தயவுசெய்து கண்களை மூடிட்டு நாடி பாருங்க. கண்கள் திறந்துருந்தா நம்மளோட எண்ணங்கள் செதறிக்கிட்டே தான் இருக்கும். டிஸ்ட்ராக்ஷன் ஜாஸ்தியாக இருக்கும். கண்ணை மூடி நாடி பார்க்க கற்றுக்கணும். ஒரு முறை இப்போ அட்டன் பண்ணி பார்த்துருங்க. ஏன்னா என்னென்ன தவறுலாம் வருன்றத நான் சொல்லுவேன். இப்போ சொல்ல மாட்டேன், செஞ்சதுக்கப்புறம் கான்செப்ட் கிளியராக இருக்கா? இதுக்கு நீங்கள் சொடக்கப்படணுன்ற அவசியம்லாம் இல்லை. இப்போ தான் பண்ணியிருக்கீங்க. பாவம், கைகளை வைக்க போறீங்க, கால்களை பின்னக்கூடாது. அதுதான் பட் பார்க்கும்போது கம்ப்ளீட்டாக ஆப்சன்ஸ் ஆகும். ரிலீஸ் பண்ணுறப்ப எப்படி வருதுன்றதை கவுண்ட் பண்ணுங்க. ஒரு 2 நிமிஷத்துலேருந்து 3 நிமிஷம் டைம் எடுக்கும். கீழே உட்கார்றவங்களுக்கு கை வலிக்குதுன்னா வேற ஒரு பர்சனை சூஸ் பண்ணிக்கலாம். நிற்காத வரைக்கும் நமக்கு பிரச்சனை இல்லை. நாடி பார்க்கும்போது நான் ஏற்கனவே சொல்லிட்டேன், மைண்டு நியூட்ரலாக இருக்கும். ஏன்னா உங்கள் ஃப்ரெண்டு உங்களை பற்றி தெரியும், அவங்க கேரக்டர்ஸ் என்னன்னு தெரியும், எல்லாருக்கும் இந்த குவாலிட்டிஸ் உங்களுக்கு தெரியும், எல்லாமே படிச்சிருக்கும். இந்த குவாலிட்டி இருந்தால் அவங்க வாத தேகி தெரியும். அங்கெல்லாம் அதெல்லாம் இங்கே வரக்கூடாது. சரியா? அதை வச்சு பார்த்தா நீங்களே டிசைட் பண்ணி தான் கை வைப்பீங்க. அப்படி பார்க்கக்கூடாது. ஓகே? நாடியில் என்ன வருதோ அதை நம்பணும் ஃபர்ஸ்ட். அம்பிளிகஸ் வரைக்கும் இருக்கக்கூடியது 8th ரீஜன். அன்னக்காலம் முதல் ஜெனிட்டல் ரீஜன் வரைக்கும் வரக்கூடியது 9th ரீஜன். ஓகேவா? அடுத்தது 10, 11, 12 கான்ஸ்டன்ட். 2, 3, 4ங்கிறது வந்து கரெக்டாக அந்த கப விரல லேட்டரல் சைடு, மீடியல் சைடு ப்ளஸ் இன்னர் ஆஸ்பெக்ட், அதாவது 3 சரிசமமாக பிரித்து 2, 3, 4ன்னு பிரிச்சு. 5ங்கிறது கப விரல் முடிச்சு பித்த விரல் ஆரம்பிக்கிறதுக்கும், ரெண்டுக்கும் கபத்துக்கும் பித்தத்துக்கும் நடுவில் இருக்கக்கூடிய அந்த ரீஜன் 5th ரீஜன். பித்த விரல்ல 6, 7, 8 வரும். திரும்ப பித்தமும் வாதத்தும் நடுவில் இருக்கக்கூடிய ரீஜன் 9th ரீஜன். 10, 11, 12 வந்து வாத விரல்ல போயிடும். ஸோ ஃபர்ஸ்ட் நீங்க பார்த்த அந்த 1 to 13 இருக்குல்ல பாடி, கிட்டத்தட்ட அதை அப்படியே படுக்க வசமா நீங்க இமேஜின் பண்ணி பார்க்கணும். ஸோ எப்படின்னா மோதிர விரல்ல ஆரம்பிச்சு இண்டெக்ஸ் ஃபிங்கர் வரைக்கும் தலை இங்க இருக்கு, கால் அங்க இருக்குன்னு இமேஜின் பண்ணி. இப்போ பிரிச்சோம் இல்லையா, 2, 3, 4, அதுதான் இந்த ரீஜன்ல நீங்க புரிஞ்சுக்க முடியும். இருக்கா அது பிக்சர் இருக்கா? நீங்க நாடு பார்க்கும்போது செகண்ட் ரீஜன்ல, அதாவது நம்பர் 2ன்னு சொல்லக்கூடிய இந்த கப விரல்ல அதுவும் இந்த ரீஜன்ல பல்சேஷன் வருதுன்னா அப்போ நீங்க புரிஞ்சுக்க வேண்டிய இடம், அது நமக்கு இண்டிகேட் பண்ற ரீஜன் இந்த ரீஜன். சரியா? உச்சியில இருந்து இளநீர் வரைக்கும் இருக்கக்கூடிய இந்த பகுதியில ஏதோ ஒரு ப்ராப்ளம் இருக்கிறது. டூல வந்து நான் உங்களுக்கு புரியுறதுனால நடுப்புறதான 2M என்ன புரியுதா? டூங்கிறது இந்த ரீஜன் தான் மொத்தமா. இப்போ வந்து நமக்கு ஜென்ரலா ரீஜன்ஸ் மட்டும் தானே பார்த்துருக்கோம். பிரதானங்கள் இன்னைக்கே சொல்றதா? பிரதானங்கள் இன்னைக்கே சொல்றதா? புரியும் சொல்லலாம் இல்லையா? சிம்பிளான ஒரு லாஜிக் தான், சிம்பிளான ஒரு கான்செப்ட் தான். அதனால சொல்லிடுறேன். இப்போதைக்கு ரீஜன் புரியுது இல்லையா? இதுல யாருக்கும் கன்ஃபியூஷன்ஸ் இருக்கா? ஏன்னா இதுதான் பேஸ். உடம்புல நீங்க அப்ளை பண்ணும்போது எந்த ரீஜன்ல ப்ராப்ளம் இருக்கு, உதாரணத்துக்கு நீங்க இன்னைக்கு நிலைமைக்கு ஒருத்தர் நாடி பார்த்தீங்கன்னா இப்போ 2ல ப்ராப்ளம் இங்கே நாடி தெரியுதுன்னா நீங்க சிம்பிளா சொல்லிடலாம் இந்த ரீஜன்ல ஏதோ ஒரு பிரச்சனை இருக்குன்றத சொல்லிடலாம். இந்த அளவுக்கு நெருங்கிட்டோம்ல, அது என்ன பிரச்சனைன்றதை இந்த நாளைக்கு சொல்லிடுங்க. ஓகேவா? இது எல்லாருனாலும் செய்ய முடியல. ஓகேவா? இப்போ 2ங்கிறது இப்போ இங்கிருந்து ஒட்டுமொத்த பகுதி இல்லையா? இதுல நிறைய இன்டெப்தா கிளாசிஃபிகேஷன்ஸ் இருக்குல்ல, அந்த அளவுக்கு இன்னும் நம்ம க்ளோஸா போறதுக்கு அடுத்த கான்செப்ட் சொல்றேன். இருக்கட்டும், சும்மா இருக்கு, அப்படியே இருக்கட்டும். போலாமா? பிரதானங்கள் அடுத்தது. இப்போ உடம்பு இந்த மாதிரி வந்து மேலிருந்து கீழ் நோக்கி 2ஆ பிரிக்கிறீங்கன்னு வச்சுக்கோங்க. உச்சியில இருந்து ஜெனிட்டல் வரைக்கும் ஒரு ஹியூமன் பாடியை 2ஆ ஸ்பிளிட் பண்றீங்க. அப்ப ரைட் அண்ட் லெஃப்ட் ஹாஃபா 2ஆ பிரியுமா? ரைட் ஹாஃப வந்து கால் வரைக்கும், வலது கால் வரைக்கும். சரியா? வலது காலில் இருக்கக்கூடிய பகுதி வந்து சூரிய பிரதானம். ரைட்ன்றது சூரியன், இடது பக்கம் சந்திரன் இல்லையா? சூரிய பிரதானம், சந்திர பிரதானம். பிரதானங்கள்ன்றது நத்திங் பட் பிளேன்ஸ். ஸோ இந்த நாடி கிளாசிஃபிகேஷன்ஸே பார்த்தீங்கன்னா இந்த டயக்னோசிஸ்ன்றது பாடி அந்த மாதிரி பிளேன்ஸாக பிரித்து தான் நம்ம பார்க்கணும். இதுக்கு முன்னாடி இப்படி செக்மெண்ட்டாக பிரித்து ஒரு பிளேன்ஸ், அதுவும் பிளேன்ஸ் தான். இங்கே நம்ம பார்க்கக்கூடிய இந்த பிரதானங்கிறது சூரியன், சந்திரன், நடுவிடம்னு பிரிக்கும். நடுவிடம்ன்றது என்னன்னு கேட்டிங்கன்னா உச்சியில இருந்து கழுத்து வரைக்கும் இப்போ என் உடம்புக்கு என்னுடைய விரல் அளவுக்கு இந்த 2 விரல், ஆட்காட்டி விரலும் நடுவிரலும். சரியா? இப்படிதான் விரல் அளவுகள் நம்ம எடுப்போம் இல்லையா? ஒரு விரல், 2 விரல், 3 விரல், 4 விரல் இருக்கும். இந்த ஆஸ்பெக்ட்ல தான் 2 விரல் இருக்கும். இதுதான் இந்த பிரமாணம். இந்த அகலத்துல இருக்கக்கூடிய இந்த பகுதி நடுவிட ஸ்தலம்னு பேர். அது நடுவிட ஸ்தலம்னு புக்ல இருக்கும். தசநாடிகள் ஸ்தலம்ங்கிறது ஏன்னா கன்னியாகுமரி மாவட்டம் இல்லையா? மலையாளம் கலந்து தமிழ்ச்சோர்கள் ஜாஸ்தி, அதனால நடுவிட ஸ்தலம்னு அதில் டிஸ்கிரிப்ஷன். ஸோ உச்சியிலிருந்து தும்பி வரைக்கும் 2 விரல் பிரமாணம், தும்பிலிருந்து ஜெனிட்டாலியா வரைக்கும் 4 விரல் பிரமாணம். இந்த அகலத்தில் இருக்கக்கூடியது நடுவிடம்னு பேர். ஓகேவா? இது முன்னாடி மட்டும் இல்லை, இங்க இருந்து பின்பகுதி வரைக்கும் இதே 2 விரல் பிரமாணம், 4 விரல் பிரமாணம் அகலத்துல வரக்கூடிய ரீஜன்ஸ் எல்லாமே நடுவிடம். அது போக மீதம் இருக்கு இல்லையா லெஃப்ட் அண்ட் ரைட்ல, இதுதான் சூரிய பிரதானமும் சந்திர பிரதானம். புரிஞ்சதா? இதுல எதுவும் டவுட்ஸ் இருக்கா? திரும்ப ரிப்பீட் பண்ணுமா இல்ல அடுத்தது சொல்லணும்? ரிப்பீட் பண்ணவா? ஒன்றும் இல்லை. இப்போ இது நான் 2ஆ வெட்டுங்கன்னு சொன்னேன் இல்லையா? அது 2ஆ வெட்டும் போது இடது பக்கம் சந்திரன், வலது பக்கம் சூரியன் சொல்லிட்டேன்னா, பட் நடுவிடம் நடுவில் ஒரு ஸ்பேஸ் உண்டு. அந்த நடுவிடம்ங்கிறது என்னங்கிறத டிஸ்கிரைப் பண்றது தான். உச்சியில இருந்து தொண்டைக்குள் இந்த தும்பி காலம் வரைக்கும் என்னுடைய உடலுக்கு என்னுடைய 2 விரல் பிரமாணம் இந்த அகலத்துல இருக்கக்கூடியது நடுவிடம். கழுத்துல இருந்து ஜெனிட்டல் ரீஜன் வரைக்கும் இந்த 4 விரல் அகலத்துல வரக்கூடிய இந்த பகுதி நடுவிடம். இது போக இடது பக்கம் இருக்கிறது சந்திர பிரதானம், வலது பக்கம் இருக்கிறது சூரிய பிரதானம். இந்த பிக்சர் பார்த்தீங்கன்னா ஸோ நடுவில் நான் ஒரு ஃபர்ஸ்ட் உச்சியிலேருந்து தும்பி வரைக்கும் ஒரு லெவல் ஆஃப் ஒரு அகலத்தில் நான் பிரிச்சிருக்கேன். தும்பிலிருந்து ஜெனிட்டல் வரைக்கும் கொஞ்சம் அகலம் ஜாஸ்தியாக. இது எல்லாமே நடுவில் தான். புரிஞ்சதா? இது போக இடது பக்கம் இருக்கிறது தான் சந்திரன், இது போக ரிமைனிங் இருக்கிறது வலது பக்கம் தான் சூரியன். புரிஞ்சதா? நீங்க யாரும் புரியாம இருக்கு? புரியலன்னா பரவாயில்லப்பா கேளுங்க. ஆக்சுவலி படிக்க தான் வந்துருக்கும். ஒருத்தருக்கு புரியலனாலும் தப்புல நீங்க திரும்ப கேட்கலாம். நான் ரிப்பீட் புரியலையா? நான் இவங்க நடுவில் கண்ணாடி போடணும் அதுக்காக உங்களுக்கு தான். புரிஞ்சதா? இதுக்கு அடுத்தது சேர்த்து சொல்லிட்டுமா? இப்போ நம்ம லேட்டரெல்லாம் நம்ம பிரிச்சோம்ல, இதே மாதிரி ஒரு உடம்ப முன்பின்னா 3 பாகமா சரிசமமா பிரிக்கலாமா? புரியறதுக்காக நான் போட்டிருக்கேன். இது உங்களுக்கு கன்ஃபியூஷனா இருந்தா நான் என்னன்றதை சொல்றேன். ஒன்றும் இல்லை, தலை கழுத்து உடம்புன்னு சொல்லிட்டு மேல இருந்து கீழே வரைக்கும் நடுவில் குறுக்க கோடு போட்டிருக்கோம் இல்லையா? என்னன்னா அடுத்தடுத்த செக்மெண்ட்ஸ்ன்றதுக்காக நான் பண்றேன். அப்படி நம்ம வெட்டுறது மேல இருந்து கீழே வெட்டுறதா மட்டும் புரிஞ்சுக்கணும். ஸோ தலையில இருந்து கால் வரைக்கும் ஆன்டியோ போஸ்டீரியரா 3 சரிசமமான லேயரா பிரிக்கணும். இப்போ தலை ஒரு அகலத்துல இருக்கு, கழுத்து ஒரு அகலத்துல இருக்கு, உடம்பு ஒரு அகலத்துல இருக்கு. அதை நான் டிஃபரன்ஷியேட்
இந்த இடத்துல ஃபார் எக்ஸாம்பிளுக்கு வந்து 9th ரீஜன்ல வந்து பல்ஸ் தெரியுது அப்படின்னா 9th ரீஜன் என்னன்னு தெரிஞ்சால் நம்ம பண்ண முடியும். அதுக்கு அந்த ஹோம்ஒர்க் கொடுக்குறாங்க. தயவுசெய்து கொஞ்சம் சீரியஸாக ஒர்க் அவுட் பண்ணுங்க. நிறைய விஷயங்கள் பார்த்த உடனே அளவு ஃபார் எக்ஸாம்பிள் தெரியாது. அது பார்த்தீங்கன்னா டயபிட்டிக் பேஷண்ட் வந்தாங்கன்னா அடுத்த செகண்டே சொல்லிடுவோம். அப்போ வேணா எனக்கு அந்த மாதிரி வந்து அவங்க டெஸ்டினேஷனாக இருக்கும். ஆனால் அதில் வச்சு நானும் லேர்ன் பண்ணேன் இல்லைன்னு சொல்லுவேன். ஏன்னா நம்ம டயபிட்டிக் பேஷண்ட் வந்து டப்புன்னு பார்த்தா நமக்கு தெரியாது. இதெல்லாம் பார்க்க தெரியும். அதை வந்து அதில் நானும் லேர்ன் பண்ணிடுவேன். அது மாதிரி தான் ஒவ்வொரு இப்போ டயபிட்டிக் நட்டுனா நீங்கள் வந்து ஒரு பேஷண்ட் வராங்க, அன்னைக்கு கூட ஒரு ஆர்கனைஸ் பார்த்தேன். நீங்கள் ஒரு பேஷண்ட் டயபிட்டிக் இல்லை, நான் டயபிட்டிக் நானே தெரியும். எக்ஸாம்பிள் பார்த்து இன்வெஸ்டிகேஷன் போட்டு கண்டிப்பாக அவங்க டயபிட்டிக்கே இருப்பான். சரிங்களா? அதனால வந்து நீங்கள் இந்த விஷயத்த கரெக்டாக என்னென்ன ஆர்கன் என்னென்னங்கிறது கரெக்டாக புரிஞ்சுக்கிட்டீங்கன்னா இந்த 13 இது வந்து பயங்கர ஹெல்ப்ஃபுல்லாக இருக்கும். ப்ராக்டிஸுக்கு அவ்வளோ யூஸ்ஃபுல்லாக இருக்கும். அது உங்கள் கையில் தான் இருக்குது. இது வந்து இன்னும் நாளாக நம்மள தான், நான் ஓப்பனாக சொல்கிறேன், நம்மள தான். ஆரம்பத்தில் நான் சசிமேலத்தை நிறையா ட்ராவல் பண்ணுறதுனால அவங்க நிறையா சொல்லுவாங்க. அதுக்கப்புறம் அதை பார்க்க ஆரம்பிச்சு தான் நான் வந்து பர்சனலாக இதை பார்த்து நிறையா விஷயங்கள் வந்து கண்டுபிடிக்க முடியுதுனால நான் அடுத்த ஸ்டெப்புக்கு கொடுப்போம். ஓப்பனாக சொல்லுவேன். அதனால் வந்து இதை நம்பி பண்ணுங்க. நிறைய விஷயங்கள் நீங்கள் ப்ராக்டிஸில் நீங்கள் வந்து ஃபீல் பண்ணுவீங்க. அதுதான் என்னுடைய அதுக்கு மேலே ஒரு சாய்ஸ்.
கிளாஸ் வந்து 4 நாளில் நாங்கள் கிளம்பி போயிடுவோம். கண்டென்ட் இவ்வளோ தான் சொல்லிட்டு எந்த அளவுக்கு நீங்கள் கேட்குறீங்களோ அந்த அளவுக்கு அடிஷ்னலாக உங்களுக்கு கிடைக்கும்.
கொஸ்டின்ஸ் நிறைய கேளுங்க. அதுதான்
கேட்கலாம் கிளாரிட்டி கண்டிப்பாக. நான் புரிஞ்சிச்சுன்னு சொல்லிட்டு நான் என்ன நான் சொல்லிட்டு இருப்பேன். சொல்கிறதுலேயே உங்களுக்கு புரிஞ்சிச்சுன்னா ஓகே. கிராஸ் வெரிஃபிகேஷன் கிராஸ் கொஸ்டின்ஸ் வந்தால் தான் அடிஷ்னல் எக்ஸ்பிளேஷன்ஸ் எங்களுக்கு கிடைக்கும். ஏன்னா நான் படித்தது 15 வருஷமாக படித்தது. இப்போ நாங்கள் சொல்லித்தரது 8 மாதமாக தான் ஆகுது. ஸ்டூடெண்ட்ஸ் ஃபுல்லாக கன்வே பண்ணுறது 5 நாளில் கொடுக்கணும் அப்படின்னா உங்களுடைய சிலபஸில் ப்ளஸ் உங்களுக்கு அட்லீஸ்ட் நாடி ஓரளவுக்கு 50% உங்களை பார்க்க வைக்கணுங்கிறது பெரிய டாஸ்க் எங்களுக்கு. அதை தாண்டி எந்த அளவுக்கு இதை நீங்கள் யூட்டிலைஸ் பண்ணுறீங்கிறது
சிம்பிள் கொஸ்டின். பூதநாட்டி சொன்னாங்க. பூதநாட்டி எல்லாருமே தெரியாது, சில பேருக்கு தெரியுதுனால. தெரிஞ்சுன்னா ஏன் ரீசன் கேட்டீங்களா? நீங்கள் ரீசன் கேட்டால் தான் அவர் சொல்லுவார். பூதநாட்டி இப்போ எனக்கு எல்லாருக்கும் தெரியாது, ஒரு சில பேருக்கு தெரியும். இப்போ ஏன் தெரியுதுன்னு கேட்டுருந்தா கேட்டிருந்தா அவர் என்ன கண்டிஷனில் தேவைவார் தெரியும் சொல்லியிருப்பாரு. அதுக்கு ஒரு ஆன்சர் கிடைச்சிருக்கும். சரிங்களா? அதுக்கு இருக்கு ஏன் வந்து பூதநாட்டி தெரியுங்கிறதுக்கு அதுக்கு ஆன்சர் இருக்கு.
இடம் பூதநாடி தான். பட் அது கரெக்டாக இருக்காது செக் பண்ணுறதுக்கு நாளைக்கு
கொஸ்டின் கேட்கறது
அதனால அந்த இடத்துல கொண்டு வந்து கேட்டுட்டு நிப்பாங்க. இந்த சொர்க்கவாசல் நாடி தெரியல பரவாயில்லையான்னு கேட்டீங்க. ஆனால் பார்க்க சொன்னது பூதநாடி, அதில் கேள்வி வரல. கரெக்டாக 2-3 பேர் கேட்டீங்க. சொர்க்கவாசல் நாடி தெரியலன்னா பரவாயில்லையா சார், பிரச்சனை இல்லையான்னு கேட்டீங்க. பூதநாடியை பற்றி ஏதாவது கேள்வி இருந்ததுன்னா கேள்வி தெரியலன்னு சொல்லி வந்தது. பட் எதுக்காக தெரியணும், எதுக்காக தெரியக்கூடாதுன்ற கேள்வி வரல. பட் நாளைக்கு எதனா நிறைய பேர் ஸ்ட்ரெஸ்ன்ற ஆன்சர் வந்தது, நிறைய பேர் கேள்விப்பட்டாங்க. பட் எப்படி பார்க்கணுன்றது இருக்குல்ல, எப்படி சார்? ஓகே சார், இப்போ நாளைக்கு மார்னிங் அந்த ஒரு ப்ராக்டிகல் செஷனில் நீங்கள் பார்த்துக்கலாம். பட் எல்லாமே எதனா ப்ராப்பராக பார்த்துட்டு வாங்க. அது நாளைக்கு 2 மினிட்ஸில் அதை பார்த்துட்டு